Hi, everybody. We have just reached the bottom of Nitinat Canyon. We are going to let our ROV pilots get set up and ready for operations today. The plan is to work our way to a slope that is just to the north of us, and we'll be working up from about 1,000 meters, or we're at 986 meters, up to around 700 meters. And hopefully finding some harm bottom and some exciting encounters today.
Definitely.
My name is Alexis Weinig, and I'm a biologist with the U.S. Geological Survey.
And my name is Paige Koenig, and I am a geophysicist with Western Washington University. And we will be your science leads for this dive. Today is our last dive of the expedition, so if you've been joining us, we've just been making our way up the West Coast as part of EX2301, Shakedown, and Express West Coast Exploration. We're happy to have you here today, and we're just going to let the pilots set up before diving into science.
Awesome. Thanks. We'll be back in just a bit with you guys. ahead, pilot. Yeah, that's perfect. Thank you. Front row, when you guys are ready, it would be awesome if you wanted to do a round of introductions also. Yeah, that'd be great. And we will, oh yeah, we'll ask video. And then when we flow through, we'll just flow into the back row.
Good morning. This is co-pilot Bobby Moore speaking from the front row. We just got down onto bottom here. Our depth today is 985 meters. And we're excited to explore up a slope today, up to about 810 meters to a second waypoint of interest. Sitting next to me in pilot today,
Sean Kennison, Team Mechanical.
and over in the far left,
Jonathan Mefford, mechanical engineer.
at nav, our video engineers, Roland Bryan and Brian Doros, are currently working on a white balance system for our main HD. If you're looking at believe camera feed 1. You see a black and white pattern, splash NOAA. That is our white balance panel. So we're currently working through some debugging on getting our main camera to white balance. And then in clipping in the back row, we have Art Howard, who helps us with our prototype still camera that we're testing this cruise, as well, while also clipping all the video and highlight images throughout the dive. We are all with the Global Foundation for Ocean Exploration, or GFOE. We're the group tasked with operating, maintaining, and piloting the ROVs and providing all the data through the satellite system and back to shore. We'll pass it over to our back row here to introduce themselves.
Thanks, Bobby. And in the back row, I am Alexis Weinig. I am a biologist with the U.S. Geological Survey, and I will be your biologist lead for our last dive of this expedition.
Aww. And my name is Paige Koenig, and I am a geophysicist at Western Washington University, and I'll be serving as your geology science lead.
I'm Caitlin Ruby, and I'm the sample data manager on this expedition, and I'm with NOAA's National Centers for Environmental Information and the University of Colorado. Happy to join y'all today.
Awesome.
Yay.
Yeah, so like our pilot said, they are getting the camera set up with this white balance system. So when we first approached the seafloor, you might have noticed that the sediment was looking relatively green. That's just an artifact of coming down and shining light into this environment. So they need to just adjust the camera optics so that we're seeing the true colors that are down here, which helps making sure that we're recording what would be, I guess, reality versus an alternate reality. And once we have that all set and ready to go, our pilots have let us know that we will make our way over to this slope that we're going to work our way up today. I think, other than that, we have been diving. We are on our ninth dive for this expedition. They start to run together after a couple days. But we are doing our second dive in Nitinat Canyon. We dove a feature that was south-facing yesterday and worked our way up a slope. Saw some really interesting geologic formations as well as biological communities. And today we're going to work our way up more of a north-facing slope. So we'll see if we see similar communities or if things are looking a little different coming from the other way.
Yeah, definitely. And as a reminder, Nitinat Canyon is offshore northern Washington. And right now we're at about a depth of 986 meters. We're glad to have you join us today.
We're going to hopefully work our way into a little bit more of a shallow area than we were yesterday. So yesterday we started at about 1,100 meters, and we worked up to around 900 meters. Today we're hoping to get up to around 750 to 700 meters, which should also put us in a different oxygen range. So one thing that our shore-based scientists noticed yesterday was that the oxygen levels the dissolved oxygen level in the water surrounding the ROV was really low. So it was around 0.3 milligrams per liter, which would indicate that we were in an oxygen minimum zone. And you get this type of environment because there's such high productivity in the waters above this deep area. So this canyon is so productive with other types of pelagic organisms. So some of these jellyfish that we're seeing, other types of fish, microorganisms that are respiring. And when they respire, they use up the oxygen that's in the water. And so that makes it difficult for some other organisms to live there. And so today we're hoping that when we get up a little higher, we will be out of that oxygen minimum zone and we'll get to experience a different type of community that might be able to use that surplus of oxygen.
Yeah, and we're also hoping with these high slopes we'll be able to observe some hard rock geology. As you might have observed when we first kind of dropped on bottom, the bottom of this canyon is highly sedimented. making it hard to find those rocky outcrops. So we're hoping for the best this dive, and yeah.
Yeah, one thing that we saw last night after the ROV was coming up to the surface that I thought was really ties into the whole how it's all connected system is there were these really cool spinner dolphins feeding off of the front of the ship. So kind of jumping, and you could see that there was a lot of them, probably
like
over
a pod of 50.
Yeah, like I was gonna say, over 2 dozen. So, likely meaning that there's healthy fish communities towards the surface. We also saw a lot of birds at the surface, which were also probably feeding or thinking that we might have been fishing and able to feed them, but indicating that these waters are really productive above us, which can fuel the productivity down here in the deep sea.
I love how interconnected everything is.
I know.
Yeah, it's fun to like observe things at depth and then just come up the staircase of the ship and observe them at the surface too. It's really cool.
And we are just outside of the Olympic Coast Marine National Sanctuary, so just to the east of us is a protected marine sanctuary, but we are working just to the west of that. But this is a great comparison for some of the work that's been done previously in the sanctuary to show that these communities extend beyond these protection zones all the way out to further parts of the deep sea. And there's often been times where as we continue to study these systems, these protections can be expanded if what we're finding indicates that that would be beneficial for management. And while we're waiting to go, we have Caitlin with us, who hasn't been able to be in the control room because she has a lot of other responsibilities on board. Caitlin, can you tell us kind of what your role is as the sample data manager and what you do when you're back on shore also?
Yeah, sure. So I'm part of the National Center for Environmental Information Data Management team. So we essentially are kind of on the back end of this data collection. So the Okeanos Explorer and GFOE work together to package all of the various data that are collected during dives and expeditions like this one. They send it down to NCEI a few months later, and then we make sure that they make it into the National Archives so that everybody who's listening in or who may be interested in this data down the line, I mean, many years, I think we save the information for a total of 60 years.
Oh, wow.
So we preserve it for long term. So if anybody wants to visit this area and see what it was like 20 years ago, they'll be able to look through our data records and watch this dive and be able to have a comparison of great quality. So I'm part of that data management team. So when I'm on shore, I'm more of a GIS expert. I lead a team that has created a bunch of really cool map products for the public to use, including a live operations map and a data atlas, which allows the public and other scientists to more easily find the information that we're collecting today.
That's awesome.
Yeah, that is awesome.
And then on the ship, I'm acting as sample data manager, which has been an exciting role to learn. So I'm just ensuring that all of the specimens that these ladies collect and these fellows collect in the front row are preserved, and all the information like what the depths were, what the lat and longs were for those samples collections. And then we do a lot of imagery of them in our wet lab on the ship. And so I pretty much organize all of that data to make sure all the bits and pieces are in one place. So if someone wants to see that sample or that coral that we collected, they can view that information via images on our data atlas portals, and then they could also request that sample in person from the Smithsonian. So there's a lot of interconnected behind-the-scene parts to data management, and I'm just really happy to be here helping you all kind of sort things through.
Yeah, we're glad to have you, especially.
Definitely.
I think there's a lot that happens behind the scenes that isn't
sure.
necessarily fully appreciated sometimes by people that don't get the chance to see that component of what we do. And I think what Caitlin was saying about archiving the data from the vehicles as well as the video is a really significant part of this operation. We often talk about where the repositories are for the physical samples that we collect. So the biological ones going to the Smithsonian Natural History Museum, and the geological ones going to Oregon State University. But there's a lot of data, like Caitlin just said, that gets stored at NCEI.
Yep.
That is also vital information that without that, these samples would have a lot less significance for the scientific community.
Terabytes and terabytes of data.
Terabytes.
Oh my goodness.
Tons of data. Yeah.
And that's something that we are continuing to work through as scientists that work in this type of environment. We collect so much data when we come down here to explore, for video data as well as sensor data, that it is always a continuous advancement of how we're going to be able to store and then access that data later on. Like Caitlin said, they have come up with a system where they're able to archive that data for over 60 years, which is amazing. And that's not necessarily always, there's not a streamlined way to do that on every expedition. So it's really amazing the work that NOAA, Ocean Exploration, as well as their partners at GFOE have put in to be able to actually create a system that all of that data is accessible to anybody that wants it.
Yeah, it's been a collaborative effort over many years to get this pipeline up and running for all these data. And it's helpful because if someone were to watch this video and I want to know the salinity throughout, the entire dive, then that's used to be a very complicated thing to find out and a difficult answer to, question to answer. And today you can definitely more easily do that.
Yeah, and I think oxygen is a really good example of something that people might want to know across the whole track of a dive. So as we're increasing in, as we are moving up in our depth range, how is the oxygen changing? And so that makes that a really great data point that's accessible. I'm going to say easily to us, but thanks to a lot of hard work by people that have programmed and figured out a way to do that for us. We're doing a quick pilot change in the front row. As we've just been able to set up, so they'll swap out, and then we will begin with our dive. And we're working in a relatively, I'm not going to say shallow for people that actually work in the shallow, but a not extremely deep depth today, so we will have a pretty long bottom time. It will only take us about half an hour to 45 minutes to ascend to the surface at the end of the dive, so we'll probably be on the bottom until around 3 p.m. local time, so that the pilots have enough time to secure the vehicles back on deck around 4:30 local time. And then we get all our samples, and we do all our other stuff.
Yep, all the processing.
Yeah.
All the photos, all the identifications.
Yeah.
So much fun.
Yeah. One of the things that we often is the case when we collect one sample of biology or geology, it might have what we call associates on it. So these are other either animals or even other rocks that are stuck to the original sample that we're interested in. And so we archive all of those associates as well. So if it's a different type of organism, we'll try and identify that organism and then preserve that in the best way separately from the original collection. So that really we might collect 4 biological samples, and we might end up with 15 biological samples by the end of the day.
Plus we take genetic subsampling as well.
Yeah.
And send those off to a separate repository.
Yeah, that's a good point, is that for every biological sample, we take a genetic subsample. So it's a small piece of the original biological sample that's preserved specifically for if someone wants to do genetic analysis, so DNA analysis on that sample. And those are sent to, still to the Smithsonian, but a different sector at the Smithsonian that has a genetic repository. It's similar to, as Caitlin was saying, archiving the data for possibly a long time. It allows us to preserve that DNA for for a long time, and so that someone might want to request that DNA 10 to 20 years from now, and it should still be decent enough quality for them to be able to sequence it on a DNA sequencer. And Paige, where do the rock samples go, and do you know what happens to them after they are on shore?
Yeah, so once we bring the rock samples on board and usually pick off the associates that live on those rock samples, we let the rock sample dry, and then it goes over to Oregon State University repository, and they have a large repository on that campus where our rock samples live. Yeah.
And is that somewhere that if a scientist needed a sample, could they go and look at all of them, or are they requesting
I believe
them?
so,
Okay.
yeah. I'm not too familiar with the repository process, but I believe that those samples are available to our onshore scientists who are interested in this region, and it's really important that we get those geologic samples, because it's really hard to identify rocks when we're just diving on a sheer cliff. We need to look at the mineral composition and the way that the minerals are oriented, and sometimes you can even take like a thin cross-section of the rock. We call those thin sections, which allow us to kind of look at the minerals in greater detail. And so that's really vital for or it makes our rock samples that we collect extremely vital for the scientists who are interested in the geologic history and how these sort of like formations are made in the places that we travel to. So, yeah.
So I've done a number of expeditions, and I always hear that it's very hard to sample the rocks that are of actual interest, right?
Yeah.
So can you tell us why that is?
Yeah, so whenever you get a rock sample, you ideally want it to be representative of the environment that it comes from, and so that usually means that it needs to be kind of plucked from the surface, and that process of kind of extracting it from the surface can be really difficult because usually the rock surface is either extremely hard and we're not able to find loose rubble to extract those rocks, or the opposite can occur, which we experienced on this expedition, where the rocks are so unconsolidated and so unstable that we're actually unable to pick it up as a whole. And so that can make it really challenging to sample these rocks with the manipulator arms that the ROV has. There are a variety of other ways that people obtain rock samples, including drilling, and just other techniques that make it easier to kind of extract the rocks from the surface.
Yeah, are those techniques normally, do they require kind of heavy machinery?
Yeah,
if you will?
exactly. They require super heavy machinery, making it kind of difficult to gather a large quantity of them, and also making it really hard to equip something like a remotely operated vehicle with
Jackhammer?
something like that. Yeah, get a jackhammer. That would be pretty sweet. Looks like we might have gotten our white balance in check.
Woohoo.
Woo!
Thanks, co-pilot.
Yeah, nice.
Or Nav, who's talking? Pilot?
Oh, yeah, and something that Jenna Hill pointed out that I forgot to talk about was that coring of like the sediment can be also really helpful to look at the layers that are beneath on the ocean floor surface, and ROVs can have the potential to take cores, and that's really, really helpful for our science in looking at the sequence of sediment accumulation.
Yeah, so some other vehicles are equipped with a like a coring system, so they either have push cores, which are about foot-long tubes that are maybe 6 inches in diameter that can just be pushed down into the sediment with a T-handle on top. And those can collect sediment that by just like essentially suction action. So the sediment is so dense that it'll stay inside the tube, and then it's brought to the surface. So all of those layers are still in the specific order that they are on the seafloor, which is really important for different types of analysis. And there's also certain vehicles that are equipped with even kind of more intense coring systems, like a vibrocore, which is something that can take cores that are over 2 meters long. So depending on what your objectives are or how deep you need to go, there's a couple different ways to achieve that. Got some of these fish.
Yeah, are those the thorny heads that we were seeing yesterday?
I think so,
those
yeah.
orange ones.
Oh, another jellyfish.
Oh, yes.
And I think that one that swam away was a sablefish, which we, I think, saw on our first dive also to the south of us. And is both of those are actually commercial species.
Oh, really?
Yeah.
Saw a lot of these thorny heads yesterday as well. I love that observation.
So we are on our way. We're going to kind of scoot our way quickly across this more flat sediment that we have landed on, which is normal for operations. We want to make sure that we bring the ROVs down in an area that we know is safe and secure for them. So we don't want them to encounter a cliff on the way down. And when we get to where we see more of a sloped region, we'll start working our way up that slope and likely see different types of biological communities inhabiting that space versus down here on the sediment.
And you can see already that we're passing through pretty dense, we call it marine snow, which is all of those suspended particles in the water column, usually indicative of high nutrient availability. Is that correct?
Yeah, I think some of this also could be like sediment coming down, so it looks pretty brown.
Yeah, it definitely does look sedimenty.
So it could be some sediment that's traveling down the canyon.
Yeah, and right now we are in a submarine canyon, which is pretty amazing geologically. So about 12,000 years ago, that was the last ice age, and about 25% of Earth's land surface was covered in gigantic glaciers. And as the climate warmed and the glaciers began to migrate towards the cooler poles, they also dispensed large amounts of water and also accumulated rock debris that it just kind of scraped up along the Earth's surface and dispense in these submarine canyons that we see today. And as there is continued water flow and also disturbance via earthquake activity, that can cause turbidites to kind of run through the canyon and kind of maintain that canyon geomorphology that was formed all of those years back.
Looks like we have another crab coming up, which we saw yesterday also. We'll see if this one looks live.
Oh, yeah. Yeah, yesterday we saw an exoskeleton.
Yeah, so a molt. Of a crab, or that's definitely what it looked like to me. We will see if that is something similar. We'll just take a quick snap zoom, pilot, if you don't mind.
And we're seeing those two red dots are kind of our measurement bars, and they're 10 centimeters across and help us measure the creatures that are in view. Sometimes the camera can be deceiving because it's so high quality that it makes these small creatures look really large.
Yeah, this one looks to be alive and
Has all of his appendages.
Yeah, so crabs are decapods, which means they have 10 appendages. So they have 4 walking legs and 2 front pinchers.
They often lose them and can regrow them in a molt.
Yeah, that's a good note. And we've seen a couple that have definitely had lost some appendages. We saw one that only had 3 walking legs left, so possibly was in a altercation. Kelly Markello from the California Academy of Scientists has let us know it's a snow or a spider crab. Kyanectes. Yeah, and Bob Carney is commenting that there was epifauna, so little organisms growing on the outside of the crab that look like sepulid worms. So those little white tubes, there will be worms that live inside.
Oh, interesting.
We have our first sea star.
Yeah, sea star. Oh, wow. Yeah, it has a lot of
I
appendages.
think this is a type of Soleaster. We'll see if Chris Mott can get a better ID for us.
Yeah, it looks like there's some jelly-ish, mucus-ish stuff hanging out on the left side of one of his appendages, which is interesting.
Yeah, Chris says Soleaster.
Okay, Solaster.
I'm just always amazed by the zoom in on this camera.
I know.
Never gets old.
So cool.
He says possibly Soleaster borealis.
Ooh, and they're predators of other critters like sea cucumbers and ophiuroids. Looks to be about 15 centimeters across.
Is that a hagfish on the bottom?
I think it's an eel pout. So hagfish don't have the dorsal fins down the back at all.
Okay.
And they don't really have pectoral fins either, so they're kind of just slithering around. Yes, that looks like a deep sea sole.
And we also saw some of these yesterday.
Mm-hmm.
Yeah, Lars, good.
Oh, and it looks like there's a sea star that's kind of on its back on the right.
Hitching a ride?
Yeah,
a little
hitching a ride.
brittle star. Yeah, thanks. So when you can hear the pilots communicating, they have a number of other screens in front of them that allow them to help navigate the vehicle. So what's projected on Stream 1 online is the main HD camera, so that's kind of the main eye for D2, but there's also on Stream 2 a view from Sirius, which is the other ROV body that kind of sits above D2, and there's also some cameras around the body of the vehicle, so on the back, so we can see what's behind us, as well as ones that project a little bit further ahead, so we kind of have an idea of what's coming. They also have a sonar on both of the vehicles, so Sirius and D2 have their own sonar systems that allow us to use sound to kind of project to see if there's any hard bodies in front of us. So you might hear them say, on the sonar you can see something is coming, and that's kind of what they're referencing.
Oh,
What do we have
what
here?
is this?
Is it an anemone growing on something?
I think so. Yeah, it looks like a snail.
Yeah,
within
because there's a trail behind
Oh yeah, and
it.
you can see
Yeah.
the foot of the snail underneath.
Weird.
With an anemone on top?
And an enemy backpack.
A bunch of hitchhikers today.
That's
Yeah.
pretty cool look.
Getting a little ride.
Yeah.
That's probably good for both of them, honestly, because the anemone has stinging cells, so we'll have kind of some level of protection, and then the snail is allowing the anemone to kind of move around and be more mobile than it would be if it was just settled on a rock.
Interesting how the anemone is facing in the direction of travel.
Yeah.
Yeah. Yeah, that's
Yeah.
smart.
Yeah.
There he goes.
And it's not necessarily a conscious decision to land on this snail, but it's somewhere that happened to be a good place to settle, and it's been able to continue to grow there.
So do you think the anemone is facing that direction because it wants to, or because there seems to be a current?
I think it's probably current. I don't know for sure.
And it looks like Annie Lindgren, who is on our science chat, has mentioned that there are a specific group of anemones that form these symbiotic associations, which is pretty cool. And also, you might hear us reference the science chat. So we have a really nice telepresence communication line on this ship where we're able to communicate with scientists who reside on shore and are interested in the location where we're diving. Since we're only able to take out a certain number of people on the ship, it's really nice to be able to kind of have that onshore science community to help us with relay that science communication about the specific site that we're at. And we are also able to do a variety of other telepresence communications, including live interactions where we can connect with communities and schools onshore via like a Google Meets line. So it's a really great opportunity to interact with the public and kind of show them what we're doing that day.
And Annie also made a comment that given the low oxygen today, so currently our oxygen level around D2 is reading 0.4 milligrams per liter. And she said given the low oxygen today, she's not surprised to see the same fish as yesterday. The thorny head has a suite of adaptations to manage low oxygen. And she thinks that the cod does also, so the sablefish. But she's not sure about that species in particular. Here's a hagfish.
Oh, okay, so that's a hagfish. I always mix them up with eel pouts.
Yeah, when you get a close-up, so hagfish have a very different, like, mouth and front end. They don't have the larger eyes, and they don't have, like, a jaw in the same way that most fish do.
Interesting. He seems to be enjoying the sediment. Oh.
I believe that's where they produce mucus. And these can produce, I think, 6 gallons of mucus.
Oh my
up to.
gosh.
And that's
there was a article a couple years ago, right, about I don't know why they were transporting a whole lot of hagfish, but there was all these hagfish in a large truck, and the truck turned over on the highway, and the whole highway was covered in slime.
Oh, that sounds awful.
So,
Not the hagfish slime.
Yeah, Jenny Waddell said the same. It covered the highway in Oregon when a truck full of hagfish crashed.
Oh, it was in Oregon.
If you Google it, that story comes up because it was a big event.
Yeah.
Ugh. And we talk about that, some mucus production with some of the sea stars also, and the corals tend to also produce mucus, especially when they're brought to the surface or disturbed. And it's thought to be essentially like a protection mechanism, so their way of saying, get away from me, stop doing what you're doing.
Black coral sample from yesterday produced a lot of mucus.
Yeah.
Oh, wow.
Black corals are notorious for producing a lot of mucus. We were able to collect a Chrysopathes black coral yesterday, and Caitlin and Ashley had the pleasure of processing that.
And then cleaning after, yeah.
Here's another eel pout. So now
Eel
you can see the eel
pout,
pout.
yeah.
Yeah,
Where it
you
has
can see the pectoral fins.
and the eye.
And the eye, yeah.
I think the hagfish just has a light sensor in lieu of an eye.
Oh, okay.
Oh, wow.
I could be mistaken on that, though.
It's also fun to look up like hagfish mouth online because they're pretty crazy looking.
Oh,
Here we go, Paige.
I'm doing it.
Compare it with a lamprey mouth
Yeah.
as well.
Yeah.
Ooh, I don't know about that. Be advised. Be cautioned.
So the eel pup is very much more fish-like in the head region.
Yeah.
Oh,
And Bob Carney
yeah.
is saying that there's a market for hagfish, especially in Korea. They usually eat them there, and the skin is also used for wallets.
Interesting.
Yeah, that's neat.
Interesting. The things you learn.
Yeah. These fish seem to like to dig around in the sediment below.
Yeah, and we're seeing the same fish occurrences. So as we go, we'll do less of these close zooms, unless it's something that looks a little new to us.
Yeah, we're hoping to make our way upslope to hopefully see some rockier outcrops.
I'm excited for the rocks, I gotta say.
Me too,
Right.
especially because the ones yesterday were so peculiar. There were so many like indentations and holes. So I'm excited to get a closer look today to see if we see the same thing on the north-facing wall.
So are we on the other side of the ridge or across the canyon from where we were yesterday?
We are across the canyon.
Okay.
Yeah, yeah. So we are in Nitinat Canyon, and yesterday we were on, I guess, the north side of the canyon, but looking at a south-facing wall.
Fair.
If you think about that direction, and then today we are on this kind of the south side of the canyon, but looking at a more northwest looking wall.
So for a canyon this size, do y'all expect kind of a similar nutrient flow downslope throughout the entire canyon, or has anybody studied that?
I don't know. That's a great question. Given the morphology of this canyon, it does seem to kind of zigzag a little bit, and the place that we were at yesterday almost appeared to be the place where the canyon kind of curved, and we call that place like a cut bank.
Okay.
And sometimes the flow, and at least in a river system, can be a lot higher, but I'm not sure if that's the same in submarine systems. So that's something I would ask the science chat if that's true.
I think that it should be the same kind of like physical aspect. I think there are some submarine canyons that have been studied pretty complexly to look at flow rates, but that's also still a question that is trying to be answered. Each canyon is specific to itself, right?
Yeah.
And Jenna Hill from USGS was telling us that when we were in Quinault Canyon, they just put out a system to study the rate of flow and other oceanographic dynamics of that canyon, and that they should hopefully have that data back next year.
Oh, exciting.
So for that canyon, we'll have a lot more information about the physical oceanography aspects, but that also will definitely inform the biological aspects. We have a different sea star here.
Ooh, interesting.
Chris Ma says, who might this be?
Who is this? Ooh, interesting.
Very spiny on the side.
Yeah, very spiny. Looks like accompanied by some slugs. Is that what those are?
Sorry?
Snails?
Oh, yeah. Some of the snails. Buconid snails. It's interesting. There's the real strong or real intense spines on the side, but we're not seeing them on the surface of the sea star like that Hypnonaster we saw.
Oh, it looks like Chris Moss sent us an ID. This is a giant mud deep sea star, otherwise known as, oh man, let's see if I can pronounce this, Therissacanthius peniculatus? Peniculatus? Pina colada. No. Just kidding. Giant mud deep sea star. I'll stick with that.
Yeah.
Apologies with my...
Very apt description there.
It's hard. So when we kind of say some of these names, they're Latin names. So they are the scientific names for these organisms, which can get us down to what specific species it is. Oftentimes we'll talk also just in common names because it's easier to understand. And sometimes the Latin names can be very long and complex. They often mean something in Latin that describes the animal. So we have certain names that mean, like echinoderm means spiny skin, and that is the whole class that contains sea stars and urchins and all of that. So they often help us to describe things, but that means you have to understand Latin.
It also looks like those deep sea mud stars usually swallow a lot of mud because they lack extendable stomachs,
Oh.
according to Chris Ma. Oh, wow.
Is this another?
Didn't we see this as in Quinault Canyon? We saw those
I
large, although it doesn't look the same,
It doesn't look the same.
but it is a large jelly of some sort.
It's hard to see under the bell, so the bell is that whole top of a jellyfish. And on
Yeah.
the ones in Quinault Canyon,
Yeah, it looks like to be about 20 centimeters across. Interesting. It seems almost to be kind of like not weathered on the outside. I don't want to say that, but looks like it's kind of been around in the sediment.
yeah.
Yeah, we have Bob Carney saying it does kind of look like he's in bad shape.
Yeah,
Likely
possibly one of those big red, the common name is a big red jellyfish.
Yeah.
Ashley thinks it might be an inside-out dead Parrelia
Oh,
jelly,
okay.
Oh, interesting.
which would potentially explain maybe the sediment or decay.
Yeah,
that we're
maybe
seeing.
lack of arms too. Yeah.
Yeah, no oral arms. That's what I noticed too, Chris, is that didn't have any tentacles projecting.
So if you all were tuning in during the descent, we saw a lot of live Parrelia on the way down today.
Yeah, we're
can
not
you-
seeing him quite. There's maybe one in the Sirius
Yeah,
cam.
one or two. Looks like we're approaching a sloped region. We've got to zoom in on a brittle star to the right and a sea cucumber
Yeah.
to the left. It's interesting to see some of these scattered shells at the base, these shell fragments.
Mm-hmm. There's a little shrimp. Oh, another couple more snails with that same anemone on them.
Yeah, a small little one.
Yeah, it looks like a small solaster. Means sun star.
They do look like suns.
I don't know if they always have I don't know.
To rely on
Chris
Chris
Ma.
here.
I know it's not an octopus.
Yeah, it looks like we're seeing more sea stars at the base of this side of the canyon as opposed to what we saw yesterday, which is kind of neat.
It's also quite different from what we were seeing more to the south when we were off of Northern California, where we had a lot more benthic organisms on the sediment. So a lot more sea cucumbers and holotherians and sea stars and brittle stars that were just kind of living in the soft sediment. Here, there's not quite as much diversity.
I feel like there's more fish, though, at this latitude.
Yeah, there are a lot of those thorny head fish.
And eel pups.
Yeah. Oh, Lars, Chris is saying that arm number can vary in the species. They can have 9 or 10 arms, depending. Yeah. Ooh, something interesting in the water column.
Jelly.
Yes,
Oh, yeah, we were seeing these on descent.
swim away.
Cute. Aw. Are those little orange things that the same thing that we like suction sampled in some of that? Because I remember seeing them in some of our samples yesterday, but I can't tell if they're the same organism.
So
The
I think
shrimp?
I think they're a type. So the ones we're seeing are a type of shrimp, but probably a different species than the ones we collected yesterday. The ones we collected were in association with the sponge collections that we suction sampled, and so I think those were smaller versions that often like live on other organisms.
I see.
Whereas these ones up in the water column are mostly pelagic shrimp, so they'll spend the majority of their time swimming around up in there.
Oh, nice.
Are shrimp typically more benthic versus pelagic?
I think there's a lot of different species. I think there's a lot of shrimp that are in the pelagic zone, so that spend most of their time in the water column.
Okay.
And there's a decent number of species that are associated with the benthic community, but I'm not an expert enough to know which has a higher diversity.
Okay. I was just wondering if one species kind of likes both or subsection.
They also tend to migrate, so they go through, a lot of shrimp go through what's called a daily vertical migration, which is the largest migration on Earth.
Oh my gosh.
So it's
really?
it's the movement of organisms in the water column from the surface to depth, and they do that depending on what time of day it is.
Oh, interesting.
So normally during the nighttime they migrate up into the surface because it's dark, so they're relatively protected. There's not that light shining. And then they'll migrate down into the deeper waters during the day to feed and get out of that light zone.
And I believe that migration is visible through midwater sonar as well.
Mm-hmm.
Oh,
And
interesting.
so in your water column sonar, you can actually see that migration throughout the day.
Yeah. There's something called the deep scattering layer that when we do like water column work or we do a CTD, we can kind of tell where that is in the acoustic data, and that will move depending on what time of day we're looking at it.
Looks like
Here's
we're looking at a scale. Is that a scale worm?
Yeah, Paige.
Yeah. Not gonna lie, I did look at the science chat. So I'm gonna shout out Kelly here. But I think that is another one of the mud. What were they? The mud. Giant mud deep sea star? Was this what we're looking at?
Yeah, so this is a
Pilot, can we get a still after you finish the
or
video?
a scale worm.
Thank you.
Oh, is that its track? Kind of those
Oh, it
dots?
looks like
Oh, that's
it.
really cool.
Yeah.
Wow.
That is really cool. I don't think I've ever really noticed that before.
Oh, interesting.
Good observation.
And it looks like that association with the scale worm and the star haven't been reported before. So just another discovery.
Thank you, sir.
Cool. Thanks, pilot. That's a real. I love that. So Chris Ma says like he's seen these in museums for years because we learned more about the biology in the last 40 seconds than I've known for decades.
Yeah, that really highlights how important this like active ROV and video work is. That's so cool.
Yeah, so a lot of historically the way that organisms from the deep sea were collected was through trawls or dredging. So running a net along the seafloor or like box cores, so sending a large coring device down and collecting a chunk of sediment. And so you didn't always know about the behavior or the ecology of an organism or a community. You just kind of knew that something... It was coming from this general area where we ran a net. And so getting down here with these really high-quality cameras and this ability to bring light with us allows us to see how some of these organisms behave in their natural habitat and learn a lot more about their behavioral characteristics versus just the physical aspect that we might learn from having a specimen brought up in a net. And we have the ability to really target our collections with this approach, so we can decide after looking at the video or having an expert on the line who can identify something, if we actually need to collect it for identification, or if the video footage is high enough quality for us to be able to determine something like species off of that video.
interesting. Oh, yeah, it looks like an anemone.
Like some
With
of the ones we were
another.
seeing yesterday.
Joined by a crab.
Oh, yeah, look at it. Just
He's covered in
With
sediment.
little legs peeking out.
And we started today at about 990 meters at depth, and right now we're at about 952 meters, so we've already migrated a little ways up slope, but we're hoping to get to a final slope of about 790, or a final depth of 790 meters by the end of our dive today. So we're already making way.
Yeah, definitely a little bit of a
Like incline, yeah.
starting to see small rock rubble or more consolidated mud rubble.
Yeah, probably likely some rock that have just fallen off from the slope, traveled their way down.
Is that another one of the soul fish?
Yeah, I think so. I think it's just a species difference, so I think this is just a different species. We did have someone ID this via email. I'll have to go back. Alan Collins had sent it to a fish expert, and he said this is a pretty common deep-sea sole, but they're not commercially fished because they're not very meaty.
Yeah, they do look kind of thin.
Oh yeah, let's take a zoom on that. It might be on a sponge.
Yeah.
It's hard to tell.
Yeah, what is that?
Are those little worms?
Yeah,
to the right
white
as
tubes
well.
are sepulid worms, and then there's a little ball sponge on the top. I'm curious. I don't know. This could be just a really grubby sponge, or it could be sediment.
Yeah.
Okay.
Oh, wow, okay.
Can we look at this little rock in front of us? Just quickly.
This is my favorite when we see this, like, random rock rubble that biology has kind of picked up on and utilized, and it almost looks like there's a bunch of creatures that have called this little rock rubble home.
Little piece of
Oh,
coral.
yeah, it does have some...
Yeah, let's take a second. Yeah, so there's definitely a small coral, and then you see those other little pink projections to the left. We have a bunch of little shrimp, so those are like what we collected
Oh, I see.
yesterday.
Couple different types of shrimp, it looks like, maybe.
Maybe,
maybe
or
juveniles.
just size classes, yeah. There's some hydroids also, so that is a little pink octocoral. It's hard for me to... And then those are capryllid amphipods, so we collected some off of one of the sponges yesterday. And they are also all over this rock, so I thought they were
Yeah.
little coral polyp settlements, but they look
Yeah.
like those amphipods.
Oh, interesting.
And then we did collect one of these shelled animals yesterday, so we thought they were brachiopods, or that's what we thought we'd been seeing all day. But when we had one at the surface that came up with one of our suction samples, it definitely looks like a bivalve. So it looked like a small scallop to us. It didn't have the morphology of a brachiopod. So there might be some brachiopods, but I think there's also some scallops down here.
I think I saw one swimming away yesterday.
Okay.
Yeah.
Oh, nice.
I don't think it's a deep sea mud star. I think it might be. Chris had said this is the one that we just saw kind of up on that what was possibly a sponge or a rock.
Uh-huh.
So it might be a Zoroastrid. They are, but
Just a little different.
it's just different.
Yeah. Interesting. It's funny how it's those two arms over on the right are so close together.
Mm-hmm.
Yeah.
Very odd looking, says Chris Ma. Thank you, pilot.
Okay, so Chris says that those older-looking, he called them Zoros or Zoroastrids, are thought to be early in the history of sea stars, so in the sea star lineage, and that particular one resembles sea stars from the Jurassic period.
Oh, wow.
Well, I mean, it's not from the Jurassic period, but it looks like ones that are.
Would it be correct to say more closely related?
Yes, or that they are at the closer to the base of the evolutionary tree. So
Okay.
they evolved that type of family evolved earlier than ones that have diverged since then.
Okay. Okay.
Another eel pout, another fish.
Woo!
Such bright coloration.
Yeah.
Yeah, they're so pretty.
Do these fish ever go into the photic zone?
I don't know.
That's a good question.
I always find it interesting animals this deep when they have coloration and an environment that other animals can't really see anything.
Right, yeah.
So it makes me wonder what the reasoning, or maybe not reasoning, but cause for that adaptation.
Right, yeah. I remember reading about those jellies that we've been seeing who are really like bright red when we shine our lights on them. The reason that they are that bright red when we can actually see them is because when they're in the dark, that red makes them look even darker than they are. So I'm wondering if maybe in the dark sea it's kind of the same thing with the fish, but I'm not sure.
Yeah, interesting. Is that a baby sole too?
Oh, yeah.
Yeah, smaller
There was a
one.
big
Really
one
small.
that we just passed as well.
No, you're right about the comment. I'm not sure. Ashley would be the one for us to ask.
Yeah, let me ask her.
So Annie Lindgren said that these thorny heads are found below 200 meters, so not much in the true quote-unquote photic zone, so where there's light, but the red coloration would be advantageous in the twilight depths. So in the depths between what we consider like shallow and deep is called the twilight zone, where you have some light coming through or some wavelengths of light, but you wouldn't have full light range.
Which is probably why the fish only have a pattern on the top and not the bottom.
Sorry?
Potentially why there's a pattern on the top of the fish and not the bottom. So
Yeah.
if something were to look up at the fish.
Yes. So a lot of them
Backlit.
have, there's a term for that and it's escaping me. They have different colorations on the top and on the bottom because when, like you said, if something's looking up at it, that white bottom will blend in with the surface above, but the red top will look relatively invisible.
Oh, wow.
Counter shading. Thanks, Annie. I know this word. I don't think so. We were asked if the soles can change color. I don't think they have what are called chromatophores, which is how they would change their the color on their skin or scales. But also not a fish expert.
I'll ask Ashley. I got a running message to her right now. Oh, so Ashley just responded to you, Lars, with your question with the long versus short spine on a fish and what that's relative to. And she mentioned long spine is if the second ray on the dorsal fin is much longer than the other dorsal fin spines. And then short spine has the second dorsal fin spine is about equal length to the other fin spines. Let me see. Yeah, let me ask. Yeah, no, that's a great question.
Yeah.
That's what it looks like.
I think that's...
Well, I don't know. There's like a little something on the bottom there, but it does look like a rock to me.
I think it's a bryozoan, which is like an encrusting animal on the outside, so
Okay.
I think it is a rock. Doesn't look like a sponge to me.
So that would be a mudstone?
Yeah, probably.
Yeah, likely it's just like some sort of sedimented rock.
Is that a scallop on the floor? It kind of looks like what we saw or what we pulled up yesterday with that iridescent.
Yeah, Chris said this sea star might be feeding.
Oh, interesting. This increase in rock rubble is making me really excited because I feel like it's leading us up to a potential rock outcrop.
There's another one on screen too.
Oh, that's pretty. Yeah.
Opening.
Yeah.
We appreciate the curiosity. It's fun. Also, Lars, one of our ROV pilots, he was asking about whether the coloration on some of those fish that we were seeing changes in whether they are male or female, and I just messaged our fish expert, Ashley, and she said that they are not sexually dimorphic, meaning that they do not change color based on whether they're male or female. Yeah. Yeah, as we continue upslope, we can see much more rock rubble, but also like some shell rubble, which we can call, sometimes referred to as shell hash. I like how these sea stars are latching onto these loose rocks. Very funny.
Interesting how they attach to a rock of similar size to them, too. They're not going to like a clump of smaller rocks.
Yeah, that's a good point. I wonder if there's some advantage to that elevation, but I don't know. Ooh. Oh. Mm-hmm. Yeah.
Sorry, I didn't have my mic on. I think this is the deep sea mud star that we were...
What are those? There seems to be like a couple, or on its right, there's this interesting, is that a shrimp?
Yeah, this is shrimp on its right. Oh, maybe this isn't, because this one has spines all the way up it. Oh, this is Hipposteria, which is ones that we saw yesterday that Chris was talking about being predators of corals. We saw this one feeding on a sea pen yesterday.
Oh, right.
Yes. That's great. Thank you.
Chris, I'm curious if you have a theory as to why these sea stars are kind of perching on these rocks. So I had asked Chris Ma, who is one of our scientists in the science chat, and I'd asked him about why we kept seeing those sea stars kind of perched on those rocks. And he just messaged and said that the answer is likely disappointing, that they're probably just feeding on stuff that's located on the rocks, either some of the encrusting stuff or maybe the brachiopods or whatever shelled sort of organisms there are. Still an interesting sight, though.
Yeah, we're definitely starting to, there's been a bit of a terrain change, just at least a little bit of harder consolidated mud pieces, so hopefully indicative of some of those at least more sheer cliff faces like we saw yesterday.
Yeah, definitely. I would say this is like rock rubble, and like we learned yesterday there are... Oh, cool. There are frequent about magnitude 6 earthquakes that happened in this region, and so those earthquakes can kind of shake up the surface and create this sort of erosion that we're looking at now.
It's another hagfish.
Don't Google what its mouth looks like.
Depends on what you think.
Well,
is cool.
it is cool. Just a little scary.
You're okay with mild horror. Yeah, so if you are listening to both the science side of the conversation as well as the pilots talking, you'll hear often a request to move the ship 30 meters. So that is the most that we're able to request to move the ship for safe operation. So we've been requesting that pretty frequently because we're kind of on the move to get up this slope. And so other dives, you might only hear that every once in a while, but this one we're zooming, if you will.
Trying to get to those rocks.
Yeah. Yeah, and for those who are kind of unfamiliar with our ROV systems, all of our ROVs are connected kind of on this tether to the ship. And so we have Sirius that's connected to the Okeanos at the surface, and then in between, or Sirius is kind of in between D2 and the ship, and then D2 is closest to the surface at the bottom.
Oh yeah, there's a little sea pen.
Oh, neat.
Yeah.
Our second coral for the day. So these are a type of octocoral that are secured in the sediment. So they have a little foot that's underneath that they submerge in the sediment and anchor themselves. And then they're normally just this one stalk, so they don't have a branching pattern. And then those fluffy parts are the polyps coming off of the stalk on each side. Certain sea pens are really tough to collect because they will retract all the way into the sediment.
Oh, wow.
So if I've attempted before and asked ROV pilots to attempt and then frustrated them because we go to either use the arm to collect or even the suction sampler and they just completely disappear. So they might be pretty heavily embedded in the sediment.
So they don't require that like hard rock to attach to?
They don't, yeah.
Okay.
So you often find these in more sedimented areas like this because they're adapted to be able to use that as their habitat.
I see.
And there's a cool group of them that is there's a cool group of them called rock pens that actually like use their peduncle as a little suction cup and they'll suction to a rock, but they can kind of hop around, so they often don't stay stationary if you go back to the same place. They have them in the shallows as well. They'll have moved. Not really understood why, but maybe just to get in a better spot.
Interesting.
Has anyone seen them move? Do they kind of just
I
detach and float
yeah,
away, or
they
do they
might.
actively kind of like worm
I don't know
their
if
way?
anybody's seen. I don't think it's a quick process.
Yeah.
So it might, if somebody set up like a time lapse camera, maybe where
Yeah.
they do this in like intertidal zones, you might be able to catch it. But I haven't seen that.
Oh.
We have some more anemones. The rock rubble's getting a little more dense.
Yeah, and I'm
That's
kind of
promising.
excited about this rock rubble because it doesn't seem to be, I mean, it's still definitely covered in sediment, but you can still see a lot of the individual rock fragments, meaning that it could have happened recently, or at least in
More
geologic time recently.
We've got a big sea star on the left. Looks similar to the ones we've been seeing.
So having more angular rock rubble means there's been less weathering of it over time. So is that how you're determining it may be more recent?
Yeah, and I would say like for the more recent, I'm looking more at like the degree of kind of that mud and silt, that finer sediment that's located on the top.
Okay.
And there seems to be a little bit, but we've seen areas where that rock rubble you like can't even see it at all because it's just completely covered. So yeah, I'm getting excited. Hopefully a rock face coming up.
think they're the Perennials
Is it really a
jellies.
snack of jellies?
So for for the folks tuning in, if you look at Cirrus's point of view, which I believe is camera 3 on there, stream 2, my apologies, then you'll see all of the jellies that we're referring to. If you're tuning in to stream 1, which is the main feed from Deep Discover, you may be questioning what jellyfish we're referring to, so just switch over to stream 2. Oh, it does.
That's a good.
Yeah, the floating lanterns.
Earlier we were talking about the importance that these video streams have for kind of looking at the species and organisms in situ, like right where they're at. And Chris also wanted to mention that these videos not only can bring us kind of some of their, or look into some of their behaviors, but also how spaced out they are, because when we trawl some of these regions, we can bring up a large number of them, but we don't know how far apart they're located within or next to one another, and so that's something that these videos kind of bring us. Thanks, Chris. Seems to be a lot of sea cucumbers in this region, or at least right here.
Yeah, as we're coming up, they've been popping up a little bit more often.
Mm-hmm.
Co-pilot, we're going to need a count of the number of jellyfish that you're seeing.
An exact number, also.
I think these sea cucumbers are from the family Letmonidae. Bob Carney has put that in the chat earlier today.
Oh, interesting.
Yeah, from Elazapodida. That's fun. Right at the bottom of the screen.
Is there an umbrella?
Umbellula or a hydroid. I don't. Oh yeah, good eye, Chris. Okay, so right at the bottom of the screen, pilot in center. There's a little coral. Can we just take a zoom on that real quick? So this is another type of sea pen. It's called an umbellula, one of my favorite words. And these have this really distinct morphology where you have all the polyps at the top of the sea pen. So the stalk is bare, and then the polyps are really large. So when we take a closer look at it in a second, you'll see there's polyps all around the center, and the tentacles are all extended. This one's pretty wind blown, or current blown, I guess. There's a wind.
Yeah, really showing the direction of the current.
Yeah.
Is it suctioned onto the ground?
So
What's this one do?
It's the same mechanism as other sea pens, where they have the peduncle buried underneath.
Mm-hmm.
You can see there's definitely probably a lot of mucus and sediment, because you can see it attached to some of the polyps here.
Right, yeah, as it moves you can see it.
And it also looks like some of the polyps are shorter than other ones, so they're not possibly like new growth on the left, or the closer side to us, but you can see all the long extended tentacles from the ones on the background.
What are those dark tubes? Oh, never mind.
You can, yeah.
What are those dark tubes?
So that's the inside of the polyp, so the internal structures of the organism. So that's where they feed and also where they digest, and all of their reproductive organs are also inside.
Nice.
Yeah, Steve Oskovich in our chat says, looks like it's taking a meal of that deep sea mucus, so it's starting to possibly ingest the nutrients out of that. Hi, Steve. Steve and I are PhD buddies from the same lab. All right, I am going to hop off and leave you guys with Paige and Caitlin. Go do a live interaction.
Thanks, Alexis. Yeah, so Alexis is leaving to do a live interaction. We mentioned those telepresence capabilities. Looks like they'll be talking with school today. Not quite sure. Yeah, school today. And since there's kind of a lot of people in mission control that'll be kind of talking during this live interaction, you may hear a little bit less from us narrating, but we'll still make sure to continue talking and narrating this journey infrequently. Oh, several crabs also coming into view. Ominous that they're facing the same way.
Is that another hagfish curled up there?
Yeah, it seems like it. I can't see its face.
I see the tentacle-looking things on its mouth.
Oh, there it is. Oh, Kelly's saying that those crabs that we just passed over are snow crabs.
Okay.
Yeah.
So a fun fact about these hagfish is they lack bones and they don't have any jaws.
Oh, interesting. That must be why it looks so squiggly, scientifically speaking.
I believe, I could be incorrect on this, but if I remember from my marine biology class, they can burrow into decomposing animals, which is what they use their mucus for, and so they just kind of eat decaying things.
Oh, interesting.
Yeah, they smell for their food. They may also be preying on small invertebrates living in the mud.
Yeah, this looks like a great place for them then.
We've seen quite a few on this dive.
Oh, this looks like the Medusa jelly that we saw earlier in the expedition. Yeah, that's a gorgeous zoom in.
I wonder if that's a broken tentacle or if it's caught something.
Yeah, at the bottom. I don't know.
Maybe it got injured when predating on something.
Its tentacles also look a little frayed in a way. So I'm curious if that has something to do with it. Looks like another sea pen, kind of by those lasers. Yeah, it looks like a soul. We've been seeing a lot of those small souls.
Oh, that one looks larger than the previous one we saw.
Yeah, it definitely does look a lot taller.
We also like to see
Oh, there's even a little mini brittle star.
A little squat lobster to the left, potentially.
Mm-hmm. Thanks, pilot.
It looks like Steve, I can't see your last name on the chat, said that that last C pin was either a, may need help with pronouncing this, funculia or a halipteris, now Baltasina.
Interesting. Those Latin names are so
I know,
rough.
They're so rough. I know I just totally butchered that. But
No, great job. Could we get a zoom in on that pink anemone over on the right? Thank you. Awesome, thank you. Yeah, this looks great. We have some scientists who are interested in kind of peering into what this looks like. It looks like this anemone kind of holds up its tentacles in a urchin-like manner. Thanks, video.
Great shot, guys.
Ooh, yeah, there looks to be a cluster. Oh, hello? Do we have someone on the teleconference line?
Hello.
Hi,
everyone. It's Jenny Waddell calling in from Olympic Coast National Marine Sanctuary, where I serve as the research coordinator.
Hi, Jenny. It's great to have you on the line.
Hi. Yeah, thanks for the invite. This is really exciting stuff for us. People who are interested in the seafloor like to see what's down there regardless of what we find. But some of these images and videos have been incredibly important for us.
Yeah. Do you want to talk a little bit about the importance of Nitinat Canyon and to the Olympic Coast National Marine Sanctuary?
Sure. Yeah, so Olympic Coast National Marine Sanctuary is a large marine protected area that encompasses about 3,200 square miles off the coast of northern Washington state. It was established in 1994 because of the incredible biodiversity of this region, but also to protect the resources because of the overlap of the sanctuary with the usual and accustomed harvest areas of 4 coastal treaty tribes that have lived along this coastline since time immemorial. This includes the Ho tribe, the Makah tribe, the Quileute tribe, and the Quinault Indian Nation. So these 4 tribal sovereign nations have rights to marine resources that extend up to 40 miles into the ocean. So the area that we're diving here in Nitinat Canyon is outside the sanctuary, but it's right near the border, the seaward border of the Makah tribe's usual and accustomed area. So we work very closely with each of the tribes to protect these habitats and resources while still allowing compatible uses within the sanctuary. So one of those important compatible uses is actually fishing. We are not involved in fisheries management here at the sanctuary, but we're certainly interested in helping to keep the habitats and marine species upon which the tribes and other fishers depend, as well as the ecosystem, to keep them healthy and robust, abundant. So learning about what's down on the seafloor is really important for us. We think about 95% of the seafloor within the sanctuary is actually unconsolidated sediments of mud and silt and a lot of what you see here. Only a small percentage is actually hard bottom habitat, and a lot of the hard bottom habitat that we see is substrate like this, where there's rocks and cobbles spread among the unconsolidated sediments.
Yeah, that looks to be kind of what we're seeing today and what we saw a little bit yesterday. Do you think that's due to the earthquake activity in the area?
You know, it's hard to say how much the tectonic activity here shapes the landscape. I mean, certainly it does, but I don't know that I would say that that's why we're seeing sort of low diversity community here.
Okay.
I think we've talked a little bit about the oxygen minimum zone, and that we're still in it. We're still getting 0.33 milligrams per liter, so I think that's really constraining the diversity that we're seeing on the video. I don't know how long you can hold your breath, but I know it's oxygen to live, and most marine species do as well, with the exception of marine mammals and turtles and things like that. So low dissolved oxygen can really structure the community quite a lot.
Right. Do you think that oxygen minimum zone, do you think we'll get out of it within this dive? Because I know we're going from about 990 meters depth to 790 meters depth. So do you think we will, or is that high oxygen availability kind of further upslope in shallower regions?
Yeah, there will be more oxygen available as we climb in the water column. I was watching the metrics from yesterday's dive, and it looked like between about 500 feet and 800 feet, the oxygen dropped pretty quickly.
Oh, okay.
And so I'm hoping that when we get back up to 800 meters, we'll see the oxygen, dissolved oxygen in the water begin to climb. And hopefully we'll see additional diversity as more low oxygen tolerant species start to fill in the gaps. You know, these guys can really tolerate some low oxygen, but there are other species that I would expect to see as we get incrementally more oxygen dissolved in the water, say maybe halibut and
Oh, wow.
some other species. But these values are really low, so I think that's really controlling what we're seeing here.
Yeah, that makes a lot of sense. I know that right now we're at about 857 meters depth, so hopefully soon we'll be reaching that kind of 800 meter threshold, and we might see a difference in diversity. Yeah, very exciting as we continue to ascend up the slope.
We're currently at 856
Yeah, it's really helpful
meters,
to.
so hopefully pretty soon.
Yeah.
It's really It's super helpful to be able to see the water conditions and the water properties. So having that sensor on the ROV so that we can understand what's happening in the water column is really helpful.
Yeah, I bet. Have you been surprised by some of the things that we're seeing at this depth? Is it similar or different to the types of organisms that you see in the shallow depths?
Yeah, it's very different from some of the animals that we see in the shallower depths, but I am a little bit surprised by some of the absence of some of the species that I expected to see based on previous dives in adjacent canyons.
Oh, interesting.
We were talking in the chat about the lack of fragile pink sea urchins, which have been very abundant in these types of habitats in the past, but we're not seeing them.
Yeah,
that's kind of interesting.
Yeah, that
how
is
different
interesting.
This canyon is from some of the other ones.
Yeah, that is fascinating. I know a couple days ago we were in Quinault Canyon further south, and even there we were seeing some different species. Yeah, so it's curious to us as well. And I bet it has to do with the oxygen minimum zone that you were talking about.
Yeah, I also think that because this canyon is out further than Quinault, I guess I'm just wondering about, distance from shore and distance from other areas that might be source populations, but just a guess.
Yeah, no, definitely. That would make a lot of sense, at least for the species distribution. Well, Jenny, we were really glad to have you on the line. I don't know if there's anything else that you'd like to add, but we really do appreciate our onshore scientists being able to call in and clue us into some of the gaps that we have in our knowledge base. So we really appreciate you, Jenny.
Thank you, and thank you so much for doing the ship-to-shore interactions with the coastal schools that we have here on the Olympic Peninsula. These kids are kind of in a remote location, and so they don't often get to have really cool experiences like this. So I just wanted to thank you guys for making sure that they get access to the cool things going on the ship as well. So thank you.
Yeah, of course. I was able to be on one of those live interactions yesterday, and those kids were so curious about what we were doing, especially in regard to the ROV piloting and things like that. So it was really, really great to connect in that way.
Yeah, we've been working hard to build up an ROV program for the schools here in Clallam County and Jefferson County. And the kids have been really excited about it. So we've had great success, and it's really so fun for them to get to see real life ROVs in action doing cool things in their backyard. So really neat opportunity. Thanks again.
Yeah, thank you so much, Jenny. I'm glad we could connect via this teleconference line. Great. So you just heard me talking to Jenny Waddell, who is one of our onshore scientists with Olympic Coast National Marine Sanctuary. Things have been a little bit quiet on our end just because we're in the middle of a live interaction and mission control, so we're trying to limit the amount of people talking. But it looks like that live interaction is just about to wrap up, so you'll be back to hearing us more often. Looks like right now we're zooming in on a
snow crab, it looks like, and I'll attempt the Latin name Crinocetes. And this looks pretty healthy, has all of its appendages, and looks like it might even have molted recently. There's not too much sediment or anything growing on its shell.
Yeah, it looks all shiny and new.
Mm-hmm.
It looks like it's also joined by one of those pink anemones that we did a zoom in on earlier. Super neat.
Getting a really nice zoom in on that snow crab now.
Yeah, I love with the zoom in camera that you can see all of the fine details that you just can't see when we're zoomed all the way out. It's really cool. It's little beady eyes. And as it nears kind of the middle of the day, I believe, are doing a little bit of a change out here in the back row to give some scientists some relief. So you might hear some new voices on the line. But the people who are jumping on are amazing, and I'll let them introduce themselves to you.
We'll do introductions right after the transition, so one moment. So as our science leads are going to grab lunch, we have a couple new folks. As a reminder, I'm Caitlin Ruby. I'm the sample data manager on this expedition. Typically, I'm just in the wet lab helping the science leads process the samples, and I work with NOAA's National Centers for Environmental Information with the University of Colorado, and we're being joined with Ashley and Jennifer. I'll let them go ahead and introduce themselves.
Hi everyone, I'm Ashley Maranzino. I am a benthic biologist supporting NOAA Ocean Exploration. I have been helping Caitlin out as a sample data manager and kind of also helping relieve our science leads during the lunch hour. But today we are also joined by Jennifer. If you would like to go ahead and introduce yourself.
Hi, I am Jennifer Clifton. On board here, I'm a mapping contractor and also a sample assistant. So I am here also helping Caitlin and Ashley process these samples that we're collecting from the ROV.
And it looks like right now we're seeing, we got some thorny heads. Let's see, what else do we have? I know earlier when I walked in the room there was a white shell that is not alive anymore, but it is indicative of this area being a former. So this area was probably an area of former seepage. So along this Cascadia margin, all along this area, there are a lot of seeps that we're kind of seeing some indication of those or past seeps in our dives and where we're mapping, which is pretty cool. And then looks like
over on the right here we may have possible stars on some of the rocks over here. Let's see.
I think they might be out of frame now, but that's okay.
But we got a beautiful fish here. Some beautiful coloration on him.
Yeah, so this is one of those thorny heads.
It's a larger one than we've been seeing.
Yeah. It
also looks like it's more color than the other thorny heads we've been seeing. Even the bottom side looks like it may be a reddish orange.
Yeah, so this is one of those, it looks like, I can't see the top spine right now, but there we go. So this is a short spine. So I know, Lars, you were asking earlier about the difference between the long spine and short spine. So the long spine has a much longer, I think it's the third or second dorsal spine is much longer, and it kind of sticks up when the dorsal spine's sticking out.
Got a little, what is that, a possible piece of a jelly that's flying across screen here?
Yep, so that's probably one of these jellyfish we've been seeing in the water column a lot, of the genus Paralia. They're all over the place. If you look in camera 2 online and you can see the view from Sirius, it just looks like a mass of dots all around D2 right now, and those are all these Paralia jellyfish that we're seeing. We've been seeing them a lot in this area. And we've got a couple other thorny heads down, so those might be the other species.
We definitely have a lot of particles here in the water column floating about. Is that marine snow, I would say?
It is, yeah. So marine snow, that's kind of the particulate we're seeing is floating down from the upper water column. And that's really what helps nourish everything in these deeper waters. So usually when we see these more productive areas, there's a lot of marine snow. That might be why we're seeing a lot of these jellyfish and a lot of different benthic organisms that are relying on some of these, all this nice carbon influx coming down in the form of this marine snow. It also looks like we've got a bit more current than we have in the past couple of dives, but I'm not positive about that.
Then possibly we may have a star here on the left with the little white arms that are sticking up here. Yes, please.
If possible, yeah, and just a quick snap. If you guys are trying to move, then you can keep on heading along too.
You can really see the texture on that sea star there.
Yeah, so we've got this nice white star that maybe we'll get Chris Ma, one of our scientists ashore, who's better able to give us an idea on this than I am. But we've got that. There were a couple of brittle stars under the rocks around us as well. You can see some of these like things that look like sticks are worms that build tubes up around them as well. And then it looks like we've got a bunch of those on one of these rocks. Little sponge. Cool, thank you guys.
What's that up ahead there, Ashley? Are you able?
Oh, the thing that looks like it's squiggling around like an eel is a hagfish. So I think we've seen a couple of those during this dive already.
Yeah,
and
we have.
Yeah, Caitlin's very excited about it, which I appreciate because a lot of people give hagfish a really bad name because they are just these like slime monsters. But I really think that hagfish are pretty remarkable animals. So they're a type of fish, but they aren't a bony fish. So they've got a cartilaginous skeleton, which is why they're able to tie themselves in knots. And they're slime monsters. They create tons and tons of slime.
They're jawless.
Yes, they're jawless. So they've got like a rasping tooth almost, rather than actual jaws with big teeth like you would think about on other fishes. So they're pretty cool organisms. Let's see, and then I think Chris Ma was asking if all of these arms right here to like kind of center frame or just to the right of the lasers. I think they are all brittle stars, but if we're in a position where we could get a quick zoom, I think that would. But again, if we're on the move, then feel free to keep heading on. Yeah, it looks like a bunch of brittle star arms poking out from underneath those rocks, and
some
then
sponges.
Some sponges, and then let's see.
What's the white pentagon star that's a little bit above the?
Yep.
Yeah.
So that little, the one that looks like a perfectly shaped sea star. It's like the epitome. So yeah, Chris Ma was saying that it's a pentagon star.
Then the fish on the right there, if I'm not wrong about this, is a type of flounder, I would say.
With
it's a little bit right to the frame. It's a little bit out of frame.
Yep,
though.
there have been some deep sea soles. Yep.
So how closely related are those to flounder?
They're in the same general order. Might be in the same. I can't remember my flounder taxonomy right now. It might be in the same family, different genera usually. So closely related, very closely.
They look similar. I've never heard of a sole before today.
Yeah, they are very similar. They're both flatfishes. They both have their eyes on one side of the head, kind of thing. Cool. That was a great zoom. Thank you guys. Awesome. Thank you. Love this hagfish just nosing its way around too.
Sniffing out whatever it wants to eat for the day.
I would also look at the sponge to the right of the star.
Yeah, and you can see all of these. So they've got a sea star in the center frame, and then all those arms that are poking out of that hole to the right of it are all brittle star arms.
Is that a glass sponge potentially in the upper
Yes.
right? A hexactinellid?
I would hazard to guess that is a hexactinellid. Great pronunciation.
Good practice on that
Yeah,
one.
kudos.
It's a very beautiful color star.
Yeah, and they're called glass sponges because of the they have a silica skeleton. It's not a true skeleton, but their sponges have what we call spicules that really build up their structure, and in glass sponges it's made of silica.
It looks like some folks chimed in the chat.
Yep, and Kelly Markello was thinking that star was also one of those Hippastrias. Yeah, and Chris Ma confirmed juvenile Hippastria. So I think Lars has gotten his biology naming down. You've gotten sea stars down.
Is that brittle star been predated on? It's missing all of its arms.
It's hard to tell if
Maybe
there's
they're
been a
burrowed?
predation event or if maybe it's got its arms underneath in the sediment. It's pretty soft here, so I'm not positive if it's just kind of digging underneath in all of this soft sediment to try and find some food.
I wonder what that shiny, metallic-y colored structure is down there.
Yeah, that's
Yeah, there's a
hard to say.
bivalve to the bottom right, too.
Oh, look at his little face peeking
Tons
out of the
life.
rocks.
Yeah. It always amazes me when we zoom in. It looks so barren down here a lot of times, and when you zoom in, then you just see so much life under every single little rock face. It's pretty impressive. Great zoom. Thank you. And for viewers back home, you're seeing these two red dots on the screen. Those are our lasers. We use them to estimate size down here, so they are 10 centimeters apart. So everything that we just saw was kind of in this area that was maybe 20 centimeters wide, to give you a size perspective. Where the zoom on these cameras is phenomenal. I think it has, 18 times zoom on this main camera. Is that correct?
That is correct.
Yeah, so that's how we're able to get these really fantastic shots, and we have all of the pilots and video. Our people from GFOE are very adept in get us these gorgeous images. So thank you for all of your
These are
zooms.
actually broadcast quality cameras that used to be used in same models as they used to use in the TV studios. So
Oh, wow.
3-chip high quality HD cameras.
Now let's look at the size of the sponge here.
Yeah, do you want to give us a size estimate there, Jennifer?
Oh, let me see. So, based off of where the lasers were pointing before, I would say since it was larger than the dots, at least in diameter, I would say it's maybe a little bit over 10 centimeters, but, once we could probably get those lasers a little bit closer to the sponge, we'd be able to estimate.
Nice,
Yeah.
yep. Yeah, it looks about 10 centimeters across to me as well. Perfect.
We'll get a zoom in on this
Oh, yep,
sponge
and
here.
Jenny's also confirming. Zoom, please. Yes, Jenny, we'll try and get you. It looks kind of like the same one that we
Got yesterday.
that we got
Collected
yesterday.
yesterday, yeah.
I know that a lot of these sponges look very similar, and it's hard to tell the species apart until you get them back into the lab and are actually able to look at those spicules underneath a microscope, and sometimes even under like a very high-powered microscope.
Even the associate shrimp look really similar to the associates we found living in the sponge from yesterday.
Yeah, exactly. So I think these are a type of pandalid shrimp as well.
I was just about to say, we can see that there's a lot of life that's also living off of this sponge.
Mm-hmm. Yep. So sponges are filter feeders, so they're able to collect all of the nutrients from that floating particulate or that marine snow that is coming down around them. So again, a lot of the other organisms we're seeing will be able to also utilize that same sort of influx in marine snow and particulate coming down. So we'll see these little oases. I don't know. Plural of oasis. We'll see an oasis of life sometimes in these areas that have a lot of nutrients.
You can definitely see the water flow or the current with the sponge and all of the organisms that are living on it. You can definitely see that there's some sort of flow that is providing a lot of nutrients to the sponge and the other organisms living off of it.
Yeah, absolutely. It's usually great habitat for sponges where there's a little bit of a current so that they can get more of the particulate matter in the water column.
Thanks for that zoom in, guys.
Yeah, thank you.
Thank you. As we're moving here, you can see all these stars and actually be able to see all the organisms that are living off of the bottom of the sediment. And I believe that's one of the fish that I was talking about earlier. That's, within the family of the flounder, yeah.
Yes, yep, so that's a deep sea sole. And it is kind of cool panning back out when when you look, it just looks like kind of a rubble field with not a whole lot of life. And then knowing that we just zoomed in and saw tons and tons of life. It's pretty cool. So probably every inch of the seafloor where we were looking is just also likewise covered in life in different forms.
So we're currently moving the ship so that we can move the ROVs. We're tethered to them, and so it's kind of like a leash system. If the ROVs want to move beyond that leash length, if you will, the ship also has to move with it.
I wonder what that is up ahead. Does that seem to be a large star right there?
Yeah, we've seen a couple of those on today's dive.
Lars, you now know more about sea star phylogeny and scientific names than I do, so very well done. Yeah, yes, exactly. Yeah, especially Chris Ma is usually the instructor. He teaches us a lot about all of these different echinoderms down here, along with the other scientists ashore in our science chat.
Yeah, those jellies are moving at a good pace.
Are we in a position where we might be able to get a zoom on that big white sea star that's coming up? Cool. Thank you.
And for those who are listening,
Lars, do you have like a notepad up there that you're taking notes on, or are you just remembering these off the cuff? There are a ton of fish and jellyfish in the Sirius cam right now. I just looked at it, so that'll be feed 2 for viewers at home. And we've got all sorts of, so there's these red jellies, and then these white, what are probably eel pouts swimming through the frame. It's pretty impressive to see that from that view if you guys are able to tune into Channel 2 at home. Yeah, Chris Ma did say this one looked a little bit different. So you were also, you also made a good note when you said, I don't know, it looks a little bit skinnier.
Oh, interesting discoloration there.
Mm-hmm.
This one definitely appears to be a lot spikier.
Yeah,
than
exactly. Yeah, I agree. Jenny says that black spot looks concerning, and I agree, Jenny. It does look concerning.
Maybe a pollinoid worm.
Mm-hmm.
That was Kelly who mentioned that.
You see all of its little tube feet reaching out there.
Oh, it looks like we might have somebody on the line. Is this Chris?
It is.
Hi, Chris.
This is Dr. Ma. Hello?
Hello, we can hear you, yeah.
Hello? Very good. So first I want to thank everybody for being so attentive. I didn't know anybody was listening. And thanks to Lars for being such a good student, even though I didn't know I had one. So this is the first time we've seen this animal. This is in the family Benthopectinidae, and this is in the genus, I believe, and believe it or not, sometimes even I have problems pronouncing these, Nearcaster. And the group's actually kind of mysterious because we collect a lot of them. The specimens come up in nets in great abundance, but we really don't think we know much about like what they do in situ. And this is an example. And, this thing comes up, it's covered in spines, and, and everybody sort of speculates, well, maybe they're suspension feeders. Maybe their spines are used for defense. And, and frankly, we're still not entirely sure what they do, but there's been a lot of speculation over the years. But, for example, this little pollinoid associate, this worm that's present in its interradius, that's a new thing. I don't think that's ever been documented before. But this is the first time I think I've ever seen this animal alive. So, and I've been studying this coast practically since I was since the 90s. So, I'm really quite grateful for being able to see it alive like this. These are, to me, almost iconic, species for this coast, especially at this depth. And, and I've seen them on deck and in museum collections, from collections that date back to the albatross. This was described in, I think, 1905 or something like that. So, the fact that everybody's able to watch it now where it lives and, surrounded by its sort of natural associates is, to me, just mind-boggling. So, anyway, I just wanted to thank everybody and to, just say how much I appreciate seeing, this is a great example of one of the many interesting species that you can see, even though they're kind of well known from specimen collections. We really don't know a lot about them. I bet that if we collected one of those rocks and brought all those brittle stars back, that there's probably a new species of Ophiacanthid there. There are so many things here that we just don't have, collections of yet or know much about. And, why are they all distributed the way they are? Because there's another one right off to the corner there. Maybe they feed on these brittle stars. It's not clear to me. So, there's a lot of unanswered questions, and these videos help answer them sometimes, but they also beg more questions. So, but anyway, I just wanted to thank everybody on the ship. I know it's the last day, but some lovely video footage, and and I'll certainly be studying a lot of these things in the months to come. So, thanks very much.
Awesome, thank you, Chris. That was fantastic. It's really good to hear that context and understand more about what we're looking at. So, thank you so much for chiming in.
Oh, it's always a pleasure. Thank you. Bye-bye.
Yep, bye.
This one is definitely a different shape than the one that we just saw.
Yes, this one doesn't have... I'm starting to learn more about echinoderms. Again, the Okeanos Bio School 101. Yes.
Less spiky.
Less spiky. It looks more inflated is how I would describe it.
It does. It does kind of look like a...
Which is probably not a bad terminology, since echinoderms have a water vascular system that they use to kind of pump through their body, so it probably is inflated.
just a little bit more than, some of the other stars that we've been seeing down here.
Yeah, so, and all of these, so the sea stars that we've been zooming in on, all these brittle stars, they're all in the that same general phylum. They're all echinoderms. So are these sea cucumbers, which look very different. So,
and then Chris just said that Ophoroid in the background looked like an Oh, Ashley, you want to help me with this pronunciation?
I can try it. Ophiomusa limani.
And maybe the species before that as well.
Oh, and then it looked like one of them was a
pedicelaster?
Pedicelaster, I would say. Pedicelaster? Pedicelaster? Yes, so if we, again,
Sorry,
you'll
folks
hear us
listening at
stumbling
home.
over names that we are not used to. Some of these Latin names are a bit rough. They
Yep.
have a lot of vowels.
Seeing them written over and over is one thing, but being asked to pronounce them.
Oh, and thank you, Chris. Chris is very good at providing us with phonetic spelling. So
Pedicelaster.
Pedicelaster. Thank you, Chris. I'm sure that it's always hard when you hear something that you're like, oh, I don't know, that's not how it sounds. So it's helpful to have context.
Well, it looks like here we almost have a trail of stars, but I want to say those are possibly brittle stars down
Yeah,
there.
we've got a ton of brittle stars on. It's just
That's an interesting change.
littered with brittle stars.
Well, it looks like the contour of the seafloor kind of changes a little bit, where it's a little bit of a slope, so it kind of looks like it
There's a
curves.
sea pen off to the left too that just left frame.
Look at all these echinoderms.
It's almost looked like they just contoured with the seafloor. You know how it moved.
Yeah, it does. Yeah, it is interesting that there's almost a trail, but it is. There's another one of those other ones that we just heard Chris talking about. Yeah, there are tons of these.
A lot of them.
Yeah. Yep, so we've got a bunch of different brittle stars. So this is, again, Chris was saying these are ophiocanthids. And then if we're able to, before we move out of this area, there was a yellow sponge just top a little bit to the right of this frame. And if we're able to get a zoom on that, Jenny Woodell says it might be a candidate for collection if possible. Yeah, that one in the top right. Yeah, so Jenny, is this, yeah, I guess we'll get a zoom in, so then Jenny, just let us know if this is something that you would like us to try and sample. Yeah, I think that sectioning would be a great idea if possible.
Would the main focus be to try to collect some of the sponge and then also gather some brittle stars that are associated to the sponge?
Seeing if the scientists want, so yeah, Abby and Jenny, if you, okay, well, let's see. Trying to get confirmation from the scientists ashore to see if they do in fact want this, but I think, are we in a position where we could do a section sample? Okay. Yeah, then I think there's also interest in some of these brittle stars that are closely associated. So if we could maybe get a little bit of a nice little slurp of kind of the sponge in a. That's what I thought too. So we might as well
collect them as well.
Yeah, again, as Chris was saying, very likely it's a possibility that some of these might be undescribed species. Perfect. And yeah, Abigail Powell was saying that this is
And for our viewers at home, we are currently working on collecting a sample. Right now we're focusing on trying to suction possibly a sponge and also some brittle stars.
Yeah. So you might hear us get a little bit quiet in the back row as we try and let our pilots focus in on that. Is it possible to, did we get a zoom on this guy before? I think we did, but is it possible to get a zoom before we suction him? Yeah, we did. Okay, perfect. Thank you. Yeah. Well, oh, perfect. Awesome. Thank you guys.
All right, thanks, pilots. That was a great collection. So I think the previous watch leads communicated to us that you guys were getting ready to make a ship move to start kind of carrying up. Okay. Okay.
Awesome.
Sounds good. Thank you.
Cool.
Yeah, it looks like we have another Hypsteria sea star possibly feeding on something. We're going to make some headway as we go up this slope. We have a lot of brittle stars here that I think just kind of started recently. Paige and I just came back from lunch.
Indeed. And in the meantime, looks like we have made a lot of headway up the Nitinat Canyon slope. We started this morning at about 990 meters depth, and right now we're at about 834 meters depth. And so, yeah, making some great time traversing up this canyon slope. We were hoping to come across some harder rock bodies, but it looks like the majority of the bottom has been silty and muddy bottom, which is fantastic for those creatures who like to inhabit this area. We've also been keeping an eye on the dissolved oxygen levels, or the amount of oxygen that's present within the water, and it seems like we're still in a pretty low dissolved oxygen state. Right now it says we're at about 0.38 milligrams per liter, and that's pretty low for this region. Is that right, Alexis? It's milligrams per... No.
Yeah, it's milligrams per liter.
Oh, okay, great.
It might not be low for this region, but it's low for most animals to be able to live.
Yeah. Mm-hmm.
And, hey, Pilot, I have a question about the current. Do you guys feel like there might be more of a current here than there was down below? Uh-huh. Yeah, it's moving.
Yeah.
Okay. Thanks for that. Okay. So, Chris Ma was saying that maybe the brittle stars might be taking advantage of that high current in this area. A lot of them seem to have their arms up in the air. These are ophiocanthids, so they have those spiny arms versus the smoother ophoroid arms.
And it looks like there's something white out in the distance. Is that a sponge, maybe?
Yeah, let's take a look. Looks like maybe one of those quesadilla sponges.
Oh, yeah.
Oh, it looks like it toppled over.
Yeah.
Right?
Yeah, definitely. It looks like that rock was kind of like set loose and took the sponge along with it.
Yeah. That's great. Thanks, video. Thanks, Pilot. Looks like we have some slime stars.
Oh, yeah.
I'm not sure if they saw those earlier, but maybe we take a zoom on one of them if you guys have the ability. Okay.
Great.
Thanks. Here we go.
Nice. This zoom in really shows you how fast the current's kind of blowing through here.
So Chris Moss says this is a pteraster, but not sure which one. It looks different from the white one that we saw the other day.
Interesting. Sounds good, pilot.
The hole in the top that we were just looking at is called the osculum. So I think Chris was describing when we looked at a different one of these the other day that they have essentially like a layer over their normal like epidermis, and so they can bring water in and out of that osculum, and that's also where they create all their slime. They're called slime stars. A lot of slimy things in this area. There's a hagfish right behind it, which also is known to create a lot of slime. Oh, we have a nice big sponge.
Ooh, nice. And also some larger kind of rock fall.
Okay, here we go.
Interesting.
Yeah, it looks like we have a coral growing on the rock that's just right of center. It's kind of a white, fluffy.
Oh, nice. Yes, Nav?
Yes, probably.
Yeah. Yeah. I think that's a good plan.
Yeah, thank you. Here we have a
Oh, wow.
stoloniferan octocoral on the right, so a white octocoral that kind of like crawls along surfaces like this. So instead of creating its own skeleton, it will attach to a hard surface like this. We've got some shrimp, some bryozoans. I think there's also a crinoid just below us. We have a pink
Is it
coral.
out of zoom? Oh, wow. Ooh, it looks like a
there's
larger
nice-
sea star, yeah.
And that's a nice crinoid right there in front of us, a feather star.
And what's a crinoid again? Is
It's
that
a type of a echinoderm, so it's related
Okay.
to sea stars and urchins, but they can be stalked or unstalked. This looks like
Interesting.
possibly. Let's see if we can get a little closer.
Ooh.
I think this is the same octocoral we saw yesterday that used to be called Swiftia and now has a new genus name
Oh.
that I need to try and remember. But we'll tag it and get back to that in our notes later.
Nice. And we got a request to look at the bottom of that crinoid, towards like the feet, if that's possible.
And the feet of a crinoid are called cirri, but that's how they kind of attach. It's definitely got some obstructions in
Yeah.
a way. There's some hydroids and an ophiocanthid that's kind of clinging on to part of that crinoid base.
Yeah. Nav, this is watch lead. Do we have an estimate to the waypoint, the second waypoint?
To the distance.
Or yeah, distance. Okay.
Thank you. Yeah, that's great.
Oh, nice. No.
Looks like we have if we have the time, there's a sponge on the right of this rock. It might be worth a quick zoom.
Oh, nice.
Yeah, with that stoloniferen underneath it. Some of those coprillid amphipods growing on there as well, or attaching the legs, kind of hanging off. So they're a type of crustacean that like to attach and feed, use the water. Look at the current going by.
Yeah, it's mega fast. Wow.
There's a shrimp inside that sponge too.
Yeah, I feel like we keep seeing shrimp inside of those white sponges.
Smart, they're smart
That's true.
dudes.
Yeah, they know where the nutrients is at.
Yeah, with this big
Oh, that's
crab.
cool. Wow.
Looks like we got another big snow crab or spider crab.
Oh, wow.
He has commandeered the sponge.
Yeah.
We'll see if we can get a look at the sponge through the crab legs. Oh, nice. It has a... I wonder if this is a really big, or similar to this, what we've been calling the quesadilla sponge. It kind of has that, like, squished morphology.
Right, yeah.
But it has that mesh above the top of the osculum, too. And I wonder, there might be some small shrimp that live inside this sponge. And curious if this crab is just using it as a perch. I don't think it would be eating the sponge, but it might be wanting to eat those shrimp that are on the sponge.
Yeah, definitely.
But crabs are normally scavengers, so they kind of go for things that have already died and they're kind of just laying around. But I think they're willing to eat pretty much anything they can get their claws on. Yeah, so we're working with a new camera on D2 during this expedition. It's a still camera where they can take snapshot images, and that's currently streaming on Stream 3, which is a Canon R3 camera, and our video engineer is working on adjusting the settings of that to take some really high quality images of this sponge and this crab. There's also seriously advanced light setup on this vehicle. So there's I'm sure maybe video can let me know how many lights we have.
Actually, there is so many lights on it, I can't.
Oh, wow.
There's so many lights that
our pilot, Bobby, he's aware of how many. He's wired them all up.
Okay, nice.
In general, we've got a lot of lights on this vehicle, and we've got 4 light swing arms, which allows us to position and rotate them to better light the subject of interest.
Yeah, so video is letting us know that the vehicle has 4 swing arms that have lights attached to them, so they can control those independently and move them to best light whatever is the subject of the image or the video.
Yeah, it looks like they're adjusting them right now.
and they are also dimmable, so we're actually increasing and decreasing some of the lights, various locations to adjust the shadowing as well.
Nice, and those lights are also dimmable, so we can kind of increase or decrease the amount of light that gets fed through the video. Oh,
and
great.
Abby Powell has said that she thinks this is one of the big quesadilla sponges. They took a sample of one of these on a different expedition and identified it as Afrocalistes vastus, which was surprising because when they're bigger they look quite different. So it's cool to see another one of these.
That pattern in the center of the quesadilla, if you will, is really cool. Thanks.
So as we're looking at this amazing crab and sponge duo, we'll just do a little reminder about what we're out here to do. So we are on board the NOAA ship Okeanos Explorer during the EX2301 expedition, so our first field expedition for the season. We are exploring off of the west coast of the U.S., so we've worked our way from Northern California up through Washington. And Paige, what have we been doing while we're out here?
Yeah, so we've been doing a couple different objectives. This is the Shakedown and West Coast Exploration Expedition, and so that means that we've been conducting a series of shakedown engineering dives, which allow the ROV engineering team to kind of set up the ROV for the upcoming field season. But we've also been conducting science dives, and we've been able to explore parts of the Cascadia margin, including some of the thrust faults and the continental shelf. And for the past two dives, we've been exploring two different submarine canyons just off the coast of Washington.
Yeah, and this is all part of a campaign known as Express, which is a campaign that encompasses NOAA Ocean Exploration, as well as the U.S. Geological Survey and the Bureau of Ocean and Energy Management, as well as some academic partners to really explore and understand the submerged systems along the Pacific coast of the United States. We've been able to collect a number of biological samples and a few geological samples. But
One.
a little harder because we're dealing with a lot of unconsolidated mudstone, so not really hard rocks, but more just hard mud.
Exactly.
But it's helping us getting this imagery is helping us to characterize what these canyons are like geologically, but also what biological communities can be found at these various depths that we're exploring.
And already we have been finding differences in the geologic environment as they relate to kind of the biological diversity. Earlier this week we were in Quinault Canyon, which had much rockier, steep slopes, and that also served as kind of a habitat for diverse coral and sponge species. And now kind of up north in Nitinat Canyon, we've been seeing a heavily eroded sediment bottom and much less of those kind of diverse species. So it's interesting to look at the connection between the geologic landscape and the biologic environment.
And we've been working alongside our partners at the Global Foundation for Ocean Exploration. They are the team of pilots and video engineers that maintain as well as pilot the ROVs and all of the video, all of the camera equipment that accompanies the ROVs. And Paige and I have been brought in as science leads. So my name is Alexis Weinig, and I am a biologist with the U.S. Geological Survey at the Eastern Ecological Science Center in West Virginia.
And my name is Paige Koenig, and I am a geophysicist at Western Washington University, which is in Bellingham, Washington. Yeah, so just like Alexis and I just mentioned, we are currently in Nitinat Canyon, offshore northern Washington. And these kind of submarine canyons are really, really amazing geologic structures. They were formed about 12,000 years ago during the last ice age, when giant glaciers covered about 25% of Earth's land surface, and the sea level was about 400 meters lower than it is today. And during that time, those glaciers kind of melted and retreated north and south toward the poles. And when that happened, they dispensed large volumes of water that kind of carved through the sediment, creating these submarine canyons and kind of these living systems that we see here today. And Nitinat Canyon is one of those canyons. Yesterday, we had a dive that was just a little bit further downslope of where we're at right now, but interestingly, we saw a lot of glacial moraine, just some remnants of those glaciers moving through the landscape. And yeah, we haven't seen any glacial moraine today, but I'm sure a lot of this sediment was derived from that glacial period.
So when we talk about the sediment being derived from that period, do we think that's the sediment that we're looking at, or is it the sediment that might be deeper underneath this layer?
Yeah, that's a great question. I would say it's likely deeper beneath this layer. As you can see, like in the water column, there's like a bunch of silt and mud already accumulating and falling down. And a lot of that sediment is derived from the continental United States that's been kind of drifted offshore, I guess you could say. So yeah, I would say a lot of that is kind of below in some of the layers beneath, which we would be able to see if we took like a push core or something like that.
There's a little rock.
Yeah, it's really interesting to see how these sea stars kind of distribute themselves onto the harder rock surfaces, but man, there's just so many of them.
Yeah, the brittle stars.
Yeah.
Here's another big.
Oh, yeah, that like
one of its arms like curled up.
Interesting.
Yeah, it looks like a sea pen. This is one we've seen a couple times and a couple small little soles, deep sea soles.
Oh, yeah.
And if you've been following along with us, this will be our last dive of this expedition. But the Okeanos Explorer will head back out to sea shortly after a port call in Seattle. to start heading to Alaska. There'll be a couple mapping expeditions to map the seafloor, as well as 2 ROV expeditions during the rest of the season.
Ooh,
nice big crab.
Hello.
Oh, there's a skate.
Oh, cool.
And a really nice squat lobster under that rock, just to the left of the skate. It has its claws out. We could take a zoom on that.
Yeah.
Yeah, and Jenny Waddell noted we're starting to see some corals at least. So there's a couple of these Paragorgia that we had seen yesterday also when we were deeper in the canyon on the south-facing wall.
Oh, there's the lobster.
It might be a crab, not a squat lobster, but very covered in stuff.
Yeah, super camouflage.
Wow, yeah.
Oh, cool.
Completely.
Oh,
so
man.
There's a hydroid growing on his rostrum, so the front. There's also some sponges growing on the carapace, it looks like.
Oh, interesting.
So those white.
He does not look like he gets punched. Looking a little dirty there. It's really weird. It looks almost like fuzzy.
Oh, yeah. It looks very, definitely has a lot of sedimentation settling on all of its little... and there's a chitin next to the skate.
Oh, wow.
We're getting a little bit more
Okay.
going on up here.
Nice. Oh, skates are so cool. And our fish expert, Ashley, has just told us that this skate looks like a female skate, and those are otherwise known as Bathiraja. And I'd be curious, Ashley, if you know what you're kind of looking at to identify that this as a female.
So it doesn't have claspers, which are reproductive organs for male elasmobranchs. They're like sharks and rays. So you can't see those, so that's a female.
Nice. Thanks, Alexis.
And we're centering this skate in the still cam, so taking some photos of it before we take a close look at it, and then we'll keep heading on. There's definitely a pretty strong current here. We can see when those fog lights were on, the sediment and the particles moving through the water, through the light, and that jellyfish moving pretty fast.
Those are great measurements.
Jellyfish float speed.
Oh, wow.
Wow.
Oh, that's super cool.
Looks like you might have had a bump on the head at some point.
Yeah. Some scars.
Wow.
Really up close and
You can see
personal.
It's just like how it's breathing.
Yeah.
So its gills are underneath, kind of where you can see it pulsing. So it's pulling water in through those openings, pushing it out underneath, through the other end of the gills, underneath the skate. And their mouths are also on the underside. nice chitin on that rock also.
Oh, yeah. Oh, wow, that's beautiful. Ooh. Right as we panned up, that crab was like, hey.
Some serious spider crabs here.
Yeah. Oh, that looks like a really platy rock right there. It's been eroded away.
Just a sneak peek. Yeah.
Yeah. Yeah, it's interesting. I kind of, I guess, started assuming as we started to see this rock debris that they would lead us kind of up toward a cliff.
Well, it might. It looks like there's some more on this side, just around that bend. There's not, and then now there's not either.
I guess we'll just keep exploring.
Oh, is that an octopus? Yeah.
Oh, it's so cute! I think I've seen...
No, there's two! Yeah, they're
Dumbo octopus! Oh my gosh! Oh man.
Yeah, it might be that they're just kind of trying to sit on the bottom. When they, if one were to take off, it'll probably have more of the mantle above it versus just being a blob.
Oh man, that's so just funny how they flatten to the surface.
So these are commonly called Dumbo octopus because they have their fins look like little ears that they sometimes will flap. I'm not sure of the species. I'm not sure of the species of these, but they are our first sighting of this type of octopus since this expedition started.
Chris Moss saying that their common name is the flapjack devilfish.
And Annie Lindgren is going to work on a species for us while we take a closer look.
Oh, great. Oh, they're so wrinkly. That's so funny. Yeah, it does look like it's melting.
Actually, this one we can't really see its eyes either.
Huh.
Oh, are they?
I'm not sure. Very strange, yeah. And it looks like the Dumbo octopus is the deepest living genus of all known octopuses. Oh, wow.
Yeah, the eyes are squeezed shut right in front of the fins, said Annie.
Oh, okay, so
those
those like white
yeah.
kind of rims. Oh, when it moves around, it looks extremely octopus-like. Beforehand, I don't know.
So they do have, even though their like skirt is kind of attached around the tentacles, they do have 8 arms. These ones just aren't as pronounced as some of the others that we've seen where they really will stretch their whole arm out. These have more of a cute little skirt.
Do they have a Oh, yeah, they do have tentacles. You can kind of see.
So they're actually
cool.
arms. So octopuses have 8 arms.
Okay.
but underneath, when we could kind of see it come up, you can see the suckers.
Oh, the suckers. That was the
line.
word.
And that's the siphon on the back.
Oh, yeah.
So that's how the octopus will bring water in and out. Oh, who do we have?
Mike Vecchione here.
Yay! Hi, Mike.
Hi, and Chris was right. These are called flapjack devilfish. It's the genus Opisthotuthis.
That's great.
I think it's Opisthotuthis californiana.
Yes.
I haven't actually looked in the literature to check, but what I think is really remarkable here is that there were two of them at the same time.
Yes.
Yeah.
Have you ever seen that
Yeah,
before,
usually
Mike?
you see them one at a time. Well, I've seen places where serrate octopods were there were high concentrations of them, so it's not unheard of, but it is remarkable to see them together like that.
And are you
Well,
aware of
there was
their
one
kind
larger
of normal
than the other.
depth range? We're at around 800 meters right now.
Yeah, that's the kind of depth I would expect to see these at.
Great. Thank you for calling in.
Yeah, thank you so much.
Yeah, well, I couldn't resist. Actually, Annie contacted me, and so I decided to see if you were answering the phone today. I tried to call earlier in the cruise, and nobody picked up the phone.
Oh, sorry.
Oh, no, yeah.
We might have had
some really nice footage of these.
We might have been on an engineering dive before, but we are in full science mode now. So we're glad you called in. And if you have any other facts or observations that you'd like to make about this type of octopus, we'd love to hear them.
Well, if you zoom in on Opisthotuthis, you'll see there's some spots that go along the arms and then up onto the head and even onto the mantle. And the actual function of those isn't known, but I suspect that they let light in under the skin, and the tissue underneath the skin is really transparent and might actually transmit the light to the eyes where it's picked up and the animal can detect shadows and such in the environment around it.
Oh, wow.
Oh, interesting.
But those little spots on the arms and up to onto the head are characteristic of the genus.
Okay.
Interesting.
I've dissected specimens looking for a structure underneath those the spots, and there's nothing there. It's just a place where there's no pigment in the skin, so it's sort of a window through the skin.
Oh, that's really strange. Huh.
And do they normally have their
Okay,
eyes kind of pulled in
enough.
like this?
I think it's responding to the light of the sub. When we first started doing subdives and seeing serrate octopods, it was really surprising that they could almost blink. They can contract the eyelid into a really tight circle around the eye, or relax it so that the eye looks really big. But I think it's reacting to the light of the sub.
Okay. I mean, that makes sense.
Yeah.
But it's also surprising. I agree. It's surprising that they can retract their eye or extend their eyelid like that.
Mm-hmm.
Yeah. You can see how gelatinous the tissue under the skin is, just because of the waves that are going through the skin as a result of probably from prop wash from the sub.
Yeah.
Yeah.
Yeah, they are amazing, and I feel like we're lucky to have gotten to see not only one, but two, and the other one looks much smaller.
Yeah, that does look...
We're kind of coming over to the other one.
Yeah, and you saw another kind of Dumbo a few days ago. It's so they're related to each other in the same family, and separate from some other families of serrate octopods.
Oh, okay.
So the one we were looking at before was
series
about...
has been pretty good for octopods.
Yeah, we have. The first one we were looking at was about 20 centimeters. This other one next to it is only about 10 centimeters. I'm going to guess that might have to do with the age of them, or do we think that they might just be different size classes?
Yeah, they grow continuously, and we don't really know how long they live. We assume it's not very long because most cephalopods don't live very long, but we don't have good ways of determining the age on things like this. So I would assume that the smaller one is just younger, but that's just an assumption.
How big do they get when they're at full, or how big can they get?
Yeah, that species doesn't get real big, so it's sort of the size of a dinner plate probably. But I haven't done a whole lot of work with the West Coast fauna, so I'm not sort of out of my depth at this point.
Okay, well, I think we're moving as the ship is moving, so we appreciate you calling in, Mike, and giving us all of that awesome information.
My pleasure. I appreciate you answering the phone.
Yeah, of course. Thanks for the insight. All right.
Okay, I'll keep watching.
Thanks.
Great. Talk to you later.
Bye.
Oh, that was a cool find.
That's fun.
Yeah, that was
Yesterday
cool.
I was complaining that we didn't see an octopus, so today we
I
got
know.
to see two.
Yeah. Nitinat Canyon was like, all right, we'll show you these two.
Looks like another octocoral as we're cruising by, which is good. We don't need to spend time looking at it.
Yeah, it's interesting seeing these like corals and sponges that have just latched onto these hard rocks. I'm curious if they have done that here or if they kind of fell down in that fashion.
Yeah, that's a good question. I think also what's interesting is some of the types of corals that we're seeing don't really grow large skeletons. So if they were to tumble and not make it or get suffocated by the sediment, we probably wouldn't see their skeleton, whereas other types of coral, if we had bamboos or even big black corals, we would see the debris from them tumbling.
Oh, I see.
But the paragorgids especially just have more of like a sclerite matrix, so a less solid and condensed skeleton. So when they erode away, they just would kind of like dissolve into the sediment.
Oh, wow.
Hey Nav, this is Watch 1. Ooh. Yeah, sorry Nav. Do you have an off-bottom time for today? Oh, now I can. Now I can. Yes. Thank you. That's perfect.
Nice. Another crab. Oh, yeah. It seems like as this slope kind of flattens out, we're kind of losing some of that rock debris that we were seeing.
So in the chat, when we were looking at the octopuses, Kelly Markello had asked is there sexual dimorphism in cephalopods, and so that means are there different sizes in the two sexes, or are females or males bigger than one or the other? And Annie Lingdon said there is sexual dimorphism in some species, but she says she doesn't think so in cerates, so the type of octopus we were just looking at. There are some swimming octopuses where they see significant dimorphism. She said in argonauts, which are paper nautilus, and blanket octopus, they both exhibit sexual dimorphism.
Interesting.
We see that with a lot of different types of organisms also. Anglerfish are one of the kind of famous groups for sexual dimorphism, where the females are really large, and they have that little angler, and then the males are really small, and they just attach to the side of the female, and they essentially just become a sack of sperm by the end of their life.
Really?
Yeah.
Oh, wow. I didn't know that fact.
So we've lost some of that rubble that we were seeing before. So it's interesting because it had to come from somewhere, right?
Yeah.
At some point.
Yeah.
But I guess we have a hard time knowing how old that rubble might be.
For sure. Yeah, definitely. And also kind of looking at the geomorphology of the canyon. When we were looking at the bathymetry earlier this morning, it kind of appeared like we were on the side of the canyon that had a couple different outlets of, I guess, kind of joining in on the main strand of the canyon. So perhaps some of that rubble is just transported through some of those outlets, but I'm not quite sure.
Oh, yeah.
Oh, neat. Ooh, a boulder. Oh, that's cool. Looks like we have a... This is exciting. What could it be?
Another boulder.
I like this boulder path.
It's definitely covered in, is that, you think, it's just sediment on the top?
Oh, yeah, definitely. It's like a carpet. Looks like some sea stars have made home there. Oh, is that a yellow coral?
That's a sponge.
Or yellow sponge?
Yeah, that's one of our lunar sponges, like yesterday. And those sea stars look a little different. So if we have the ability to zoom, cool. Nice.
Oh, yeah.
These are very different.
Interesting.
Some type of Ophiocanthid, I think, still. But we haven't seen, and they're all on this one rock.
Oh, wow.
Or maybe not an Ophiocanthid. I don't know. We'll see. Chris
Yeah, we'll
Haas
see what Chris
in the
says.
chat. We'll see what he...
Oh, yep.
Oh, he said it is an ophiocanthid.
Oh, some tiny tunicates.
And that one of those lunar, we're calling it a lunar sponge yesterday, which we collected from the other side of Nitinat Canyon.
Oh, wow.
It has a very distinct green color, which is kind of interesting. We don't see a lot of green. It looks a little yellow on screen, but on the surface it's very much like an olive green that is pretty uncommon for seeing in the deep sea. But sponges are interesting and strange, so it's not too surprising that a sponge might be a unusual color.
Oh, yeah. A solus.
I think it might be one of the... Oh, that little guy right there.
Oh, is that one of the juvenile thorny heads
I
that we were
think
seeing?
so, yeah. But that's okay. We can take a quick look at it.
Yeah. Its fins are so large compared to its body. That's funny. Yeah. Well, let's see how big he is. Oh, yeah, only about 7 centimeters or so. Maybe less, 5. Super tiny. It's cute. I think these 3 boulders are likely some of that glacial erratic that I was kind of talking about earlier, transported during the last glacial maximum. Nice. Oh, nice. Oh, yeah.
Oh, yeah.
I don't know.
Yeah, we've got a nice couple set of boulders here.
I don't know what that... Yeah, that's strange.
White object.
Interesting.
Likely coral or sponge. Looks like we have some coral. We'll take a closer look. Oh, look at all those bivalve, or like shells.
Oh, yeah, interesting. Huh. Beautiful pink anemone.
Yeah. I mean, they're not embedded, but I bet you could pick up some of those rocks.
Yeah, definitely could. I guess we could get a sample, but I think some of this has just been transported by glaciers, so I don't know how representative it would be.
Yeah, Jenna Hill in our chat made a comment that these boulders are very rounded. They really are.
Yeah, they're super
Definitely
rounded.
Been
Weathered. Oh my gosh, there's so many shrimp on the coral.
Oh, yeah. So this is a type of primnoid octocoral.
Oh, and there's a...
And it definitely is a host to a lot of these shrimp and brittle stars that seem to be using it as a place to kind of perch off the seafloor. These look like these little pandalid shrimp. We collected a couple smaller ones yesterday when we suction sampled a couple sponges, but they are really liking this.
Yeah, they are all over it. Oh man, seeing some more of that fast-moving current.
And these ophiocanthids are like all on the base. They're really not on the coral at all. They're just kind of on the surrounding rock.
Kelly said this looks like a shrimp party. Indeed, it does. Oh, that's a good note, Jenny. It looks like they're kind of found a nice spot that's protected from trawlers.
Also brooding eggs. So you can see the eggs in some of the shrimp on the underside of their body, the like kind of darker color. So see that one in the back that we're focused on? On the left top center.
Oh, in the middle. Yeah. Oh, yeah, it's almost like kind of like a pouch sort of thing.
They keep them underneath their abdomen like that, and then when they're ready to release, they would just release the eggs, and they will be planktonic little larvae until they grow into larger shrimp.
Oh my gosh, yeah.
There's a lot. Look.
Yeah, you can see like all the individual ones too. That's cool. Yeah, a lot of them have that too. Whoa.
It's like a shrimp nursery.
Yeah.
And they almost all seem to have, I'm trying to see if there's any that don't have eggs, so that would indicate that almost all of these are female shrimp, which is kind of interesting that they would all be aggregated like this. It's probably a place that they have kind of figured out is relatively safe, so the coral is not going to harm the shrimp in any way, and they'll probably be a pretty consistent food source because the coral has grown somewhere where there's high enough current for the coral to continue to feed. So the shrimp would be able to utilize that same current for snatching critters out of the water.
Yeah, and I'm sure being wedged between those two boulders also really helps for protection.
Absolutely, yeah. Don't see eggs on this shrimp.
Interesting.
So we see a couple those Ophiocanthid arms too, really up close. This has been our first primnoid octocoral for this dive, so that's
interesting.
Really nice too to see. We saw, I think, the same species yesterday, at least once on our dive. And these boulders seem to be pretty stuck in place, right? So those I don't think are rolling down this canyon anytime soon, which probably is some of the reason that those corals have been able to grow there. It looks like we have another one. We'll take just a quick snap and see if it's the same. Yeah, it looks like the same species. Cool. Also kind of perched on the side, protected by those other boulders. Oh, we got a little Paragorgia in there, too. It's cool how they grow in that formation that's kind of semicircle. I think that has to do with them oriented to the current flow. So they're probably facing in the way that they're receiving the most current flow and growing in that direction. Looks like two different kinds of crabs.
Oh, kind of facing off.
It's a big sculpin.
Ooh. Oh, yeah, that does look different.
I think those are the ones we were seeing when
yeah.
But they are really, I mean, it's interesting. They're always kind of like half on those rocks. They look like they're like half in the sediment.
Yeah.
On the underside of those rocks. I was just physically doing that in the control room.
Physically putting one leg in the sediment, arms up.
So we are headed towards a target that in our backscatter data, which is a way of detecting the hardness on the bottom of the seafloor, showed a really high return, which means that it looks like this area that we're headed to had potentially harder surfaces than what was in the surroundings. So that's why we're continuing to really move towards this target. I think we're about 50 meters away to see if that really is something significant or if it was our data messing with us.
Yeah, it could also be like a harder rock body that would make that sort of return, but never know.
I think that's a fish. Never mind, sorry.
I also saw what you saw, though, that little movement.
This is definitely like a echinoderm, sea star, brittle star haven.
Yeah, thanks to these rounded boulders.
We've got a sponge that's white right there in the center.
Is that a vase?
Yeah. These fish are definitely getting larger, too, I think.
Yeah, I have been noticing that.
Is the crab eating something?
That white thing?
Crab's got something happening. We'll take a look.
Huh.
I'm hoping he's eating this other crab. I'm a big fan of predation events.
Oh, it does look like another crab, though. Interesting.
Wow, that's a big. So Kelly Markello in our science chat is saying that these are both likely lithoided crabs. So these are in the king crab family. We will see what's happening as we set up and try and image this interaction.
Oh, uh-oh. I also can't tell if it's the lighting, but they're different colors, right?
They are different colors.
Okay.
So right now they're imaging with the still camera, and then we'll come around and take a better look with the HD. Ooh, let's.
Ooh, uh-oh. Uh-oh.
Whoa.
Oh, no.
It might be, but this crab, so the bigger crab is grabbing the arms or the claws of the smaller crab.
And it almost looks like that small crab is like retreating
It
or
is.
like
It has
looks
all
kind
of
of
its
scared.
legs pulled in.
Yeah.
It's also, one is very much a white color, and the other is like a dark red color, but they definitely look to be the same, from the same group of crabs.
Mm-hmm. This is strange.
So for some crabs, they normally mate in a very different orientation. So this is not how I've seen crabs mate before, which is kind of throwing me off for why they might be doing this. He's just taking
I don't know what's going on. You're coming with me. Huh.
Wow.
This is really interesting.
We'll see if we have anybody else chime in that might have more insight as to why they're behaving this way, but it's definitely an interesting behavioral observation.
Huh.
Wow, look how big the steps this crab can make.
Yeah. He's in a hurry for sure, wherever they're going.
Yeah, Chris asked if, are we sure that the white crab was alive and not a molt? So I think it is alive because when they started to move, that white crab really it retracted all of its legs. So I think you can see its antenna moving also. So it's alive. And Kelly Markello said she knows that some male crabs will guard females if they're about to molt, so they can be the first to mate, but not sure if this carries over into lithoided crabs.
Huh.
And I don't know if
Interesting.
in lithoidids if they display this lighter white carapace coloration before they molt. And so it could be a situation where essentially a male is taking claim to a female, but I'm not sure.
Interesting.
Seems a lot of our scientists are stumped. But that was great footage. Thanks, Violet.
That was really spectacular. And now I can't quite see if he's still latched on.
The other one is see his legs are moving?
Yeah, it's definitely out again. Maybe it just needed a ride.
If I was a crab and I saw a big thing like D2 coming with those big arms, I'd run too.
That's true, Roland. Roland just mentioned how if he was a crab and saw D2 that he would also run away.
We're doing some imaging with the still cam. Our video clipper has a really nice view of both of these crabs in his view of the camera. Yeah, let's let him go.
Yeah, sounds good.
I just love this grabbing onto the...
Also, pilot, this is Watch. I think that we would like to get one of these as a rock sample, one of these rocks. I don't know if that's possible, especially because we're so close to our waypoint, but that would be great if we could. Yeah, any of those like dark colored ones look great. And so if you've been following us during our expedition, we've been kind of along the Cascadia margin, which is a highly sedimented area. Pilot, this is Watch. If we could also get one that has one of those like spiny brown ophiurides, that would be great. I'm trying to find one that would be good. That's all right. I say that we can still try if that's okay. It looks like there's a nice one kind of to the right of your arm that's kind of latched on in the backside. Yeah. Yep, that one looks great. They all look great, so whatever is easiest to you, I would say. Perfect. Oh, yay, cool. Sweet. Thank you so much, Pilot.
And it looks like we were able to get that darker brittle star that was requested.
Woohoo!
So when those come up, we'll quickly get them into some water before we preserve them.
Yes, definitely. And if you've been following along at our expedition, we've been heading up along the Cascadian continental margin, which is highly sedimented and loose and unconsolidated. So trying to find rock samples along this margin has been really difficult because a lot of times when we try to pick them up with our manipulator arms, they just crumble. And so this was our first official rock sample of this expedition. So I'm super excited. We were able to get a different rock sample a couple dives ago that was an associate with a sponge. Oh, no, it was a, was it a sponge?
No,
I forgot.
You can do it.
An anemone.
No.
What was it with? I can't remember. I can't even visualize it.
Corallimorph.
Oh, it was with the corallimorph. That was it. But it was an associate, so it was kind of a secondary, kind of latched on to a separate sample that we gathered. Ooh, got another skate.
Oh, yeah. There's second or third. Oh, this one is a male, so it has those claspers on the back.
I see.
Some more boulders.
Oh yeah.
Wonder if that target, or is the target in Sirius possibly waypoint 2? Is that in range now? Okay.
Okay.
Cool.
Nice. Oh, whoa, yeah, there is a large.
Where?
Is that?
Oh, on the top of the rock? Is that what you're...
Oh, there.
Yeah, I think it is a hermit crab.
Oh, an interesting sea star over there, too. Oh. It's cute.
Yeah.
Mm-hmm.
Looks like a little... I think Pagiridae is the family of hermit crabs.
Interesting.
Got a sponge.
Oh, nice. Again, growing kind of on the side of the rock.
Oh yeah, I see something in the distance.
Yeah. Some more rocks. Some clusters of these large boulders. Yeah, another giant fish.
Yeah.
Looks to be about, I don't know, 40, 50 centimeters.
Yeah, maybe
across.
like 40-ish.
Yeah. Dang.
Much bigger than the ones we were seeing down below.
Mm-hmm.
These brittle stars that are on the underside of these rocks, crazy.
Yeah, they really like it there.
Got a couple more of these lithoid crabs. And that nice big sponge over there.
So floofy.
Oh, there's another big
Oh, man.
So Ashley had said that thorny heads could be commercial, so that makes more sense to me now that we're seeing bigger ones.
Right,
Because
yeah.
I was like, are people like these little tiny ones? Or?
Yeah, it'd be a lot of bone to pick out.
Yeah.
Looks like we're taking another still
You
shot.
can really see in the stream one, just the amount of stuff in the water column and how fast the current is moving.
Yeah. Yeah, it's feeling, looks like it's like coming right at us.
Yeah.
Yeah. Cool to have like a bird's eye view of the landscape a little bit.
big sea star.
Oh, yeah, whoa.
Sprawled out there.
Yeah, something that we've been following kind of as we've been going up this slope in Nitinat Canyon has been the dissolved oxygen levels, and we've been thinking as we've been like rising to a shallower depth that those dissolved oxygen levels would change, and right now it's still fairly low, which is kind of interesting. We're at 0.35 milligrams per liter, which is similar to kind of where we started at the base of the canyon.
Okay.
Okay.
Yeah,
sounds
good.
good. This is interesting. A mound of some sort with... Are those brittle stars? Yeah. Interesting. Huh.
Yeah, it's just like a mound of sediment on the top of
Yeah,
this.
that's really strange. It's like localized uplift is kind of weird. Huh. Yeah, I'd be curious to kind of see that. Deep sea, deep sea race. It's just for D2.
Yeah, it's like it'll be interesting to see, like, how it continues to, is it another, like, mound just at the top?
Yeah, I'm not quite sure. And the rocks that are kind of distributed next to these mounds are really weird, too. So I'm excited to get a better look at some of them are kind of broken and... Yeah, Jenna Hill said one of them looked like it was one of the rocks was cleaved in half. I noticed that one, too.
Yeah, sounds good.
Okay. Yeah, there was some on the right that if we could just do a quick zoom over in that direction.
This is a Nephtheid octocoral, so one that we were seeing yesterday. Meredith Everett was able to ID it to species.
Oh, so pretty.
Yeah, they're really lovely. So they don't have an internal axis like some of the other ones we see, so they're very gelatinous. And they have just internal sclerites that give them some hard structure and some ways to identify them based on species.
Nice. Cool.
Cool, cool, cool.
Cool. Okay, cool, thanks. Yeah. Is there a way to, like, back up? Is that a weird request? Okay, great. Cool, yeah,
like
that's the rock that we were talking about. Yeah, it's, like, fully split in half, which is just kind of weird. I don't know if we could get, like, a zoom in there. Cool. Yeah, it's really strange. It looks like it was cut, like, perfectly in half, and I can't really tell whether it, like, how that happened. But maybe as it fell, it just cleaved, or maybe these are two different rock bodies, but the inside, the lighter, like, minerals on the inside are kind of telling that it likely kind of opened up from the inside and is revealing kind of those less weathered surfaces, which is just strange and leaves me with more questions than observations.
Got some crinoids latching on there also.
Cool, thanks Violet for that zoom in.
Somebody came down and did a karate chop.
Yeah, that's exactly what it looks like. But why?
Yeah,
they needed room
strange.
for their motocross track, I guess, or whatever. But, okay, yeah, weird. Yeah, we're clear, thanks.
Oh, something large just swam over us.
Yeah. Ooh. Gigantic.
Another one of these big.
Ooh.
I think they're still the thorny heads.
Yeah. Oh yeah, whoa.
stir up some mud for us and
Yeah.
get on his way.
Thanks, sir. Thanks a lot. Oh, I don't know if I'd bother that one. I don't know if I'd bother a crab.
Got a fish party.
Mm-hmm. Yeah, it looks like all 3 different types just in view. Huh. Okay. Oh, interesting. Interesting topography. Huh.
So we've kind of worked our way up this slope throughout the day and haven't really seen too much hard rock or even like sheer faces like we were seeing yesterday. As we were getting towards this local high, we started to see some more of these boulder fields that I think Paige was saying could possibly be the result of glacier receding.
Yeah, glacial deposits.
Glacial deposits.
Mm-hmm.
Hey pilots, if we're at a spot at one point, would we be able to maybe get a zoom on some of these? They're a white sea star. I think we're out of view of one. I'll let you know if I see another one. Sorry. I think so, but I think they've been popping up every once in a while, so I'll let you know if I see one pop into frame. Yeah, thank you. I'm going to so we did a little bit of a changeover in the back row. I'm giving Alexis a quick break back here, so I'll let her decide where she wants the water samples. She should be back very quickly. So I think maybe that there's a couple of these white stars coming up in frame above. It looks like there's one on the left, then there's like a pink one kind of dead center, and then there are three to the right. If we could get a zoom on one of those white ones, that would be great. Thank you. Maybe to the right, that way we could also get a look at that the pink one too if we have a chance. Thank you.
Current seems pretty intense up here.
It is always interesting to see. I feel like organisms really like a little bit of current, but then sometimes it's too much, so that might be why we aren't seeing as many like sponges. We might not be seeing corals. I also think this area might be trawled, and so that might also be why there's not as much benthic coverage. Awesome, thank you. I think that's probably good on the pink one, but if we have time to go to one of those white ones, Chris Moss sounded interested in that one. Yeah, Chris is saying that that pink one is a Hippastria.
I feel like we've seen quite a few of those on this dive so far.
Yeah, I think they've been kind of common over here. That poor little thorny head just swimming against the current.
And is that a snail on the
that
top
snail
right?
to the right of it. It's interesting color on it.
Yeah.
This looks like one of the ones we were seeing
earlier. It's also very inflated looking.
Yeah, but look at how it is the current is so strong.
Yeah.
Yeah.
Yeah, for sure.
I'm surprised that it can. Is it possible to collect this guy, or are we in an okay position? Okay, awesome. That would be great. Thank you. On this guy, it looks like it might be pretty fragile. Do you think it would fit in the section? Okay, thank you. Let me double check with Chris Ma, make sure that that's a good one.
They do look like it has jello arms.
Yeah, Steve Oskovich in the science chat said jello arms, and yes, Steve, I agree.
Whatever method for collection that the pilots think is most appropriate sounds good to Chris. Can we put some lasers on to get kind of a size estimate on this? Okay, so maybe 10 to 15-ish centimeters across. Yeah, Kelly, it does look like if it let go of the surface, it's just going to blow right away. Wow, that's really interesting, Chris. So in the chat, we're seeing Chris Ma said that he's realizing the last time this species was collected from Washington was 1889. Yeah, no pressure, guys.
And he says it's an Amphoraster marianus.
Sounds like that'd be a very interesting genetic comparison.
Yeah. Well, I don't know that they would have. Whatever they collected from then won't be able to have any genetics done off of it because the advent of DNA and doing any of this sequencing is so new relatively compared to those old collections. It's probably, if it is still in preservative, it would have been thrown in formalin, and that degrades all DNA very quickly. And I don't even know if sometimes echinoderms are dried.
Yeah, I was just about to mention it may have been dried.
Yeah, exactly.
But that kind of goes to show you the importance of long-term preservation in these specimens and data collections in general, because the advent of new technologies may allow future generations to relook at these specimens and do all sorts of new analyses that aren't currently possible with our current technologies. Wow, what a cool story, Chris. So in the science chat, Chris Ma kind of elaborated on that I don't want to say most recent collection, but the last collection from 1889. It was dried and collected by the albatross. 28th of June, 1889 was the last time this specimen was collected in Washington waters. So cool to know the history of specimens.
Yeah, sponge and some more brittle stars.
But it's been really working out with this current. All right.
Awesome. Thank you guys so much.
Great specimen collection, guys. Thank y'all.
Yeah. Chris, this has been a great collection for brittle stars today. We've got brittle stars, and now we've got you an amphoraster. It is interesting that there aren't very many that have been collected in this area, because it seems like there are a lot of them if these are the same species. So that'll be a really great new addition to the collections. So all of these biological samples do get sent to the Smithsonian Natural History Museum, and they can be accessed by members of the community. Then all of the geological samples we collect get sent to the repositories at OSU. So again, it's pretty cool that we're able to get scientific input. And then I think we're okay if you guys are on the move. Yeah, thank you though.
Oh yeah, that looks like a skate, kind of buried in some sediment. Maybe something on its back. Oh.
I think it. It's not a skate.
Okay. Yeah, I'm seeing that after closer examination. I'm like, oh, maybe that is just a rock. Yeah.
You're just so in love with the biology now that you're turning rocks into biology.
Honestly, though, honestly. Well, it's also just an interesting shape. It's a very skate-like shape, I would say. Yeah.
Yeah, so there is, like, we've got, it looks like some hydroids, and then we've got a crinoid sticking to some hydroids. So the crinoid is that that yellow thing kind of sitting in the center, and then it looks like there's a little crab at the base. It does look like there's a scallop. Cool. It's a pretty cool zoom. Thanks, guys.
Yeah. Yeah, and yeah, this landscape is so deceiving, especially when it's kind of eerie and sedimented like this. It's very curious, each object. Really interesting seeing that white rock right next to that super dark rock, indicating that they're likely different lithologies or weathered in different ways. Can't quite be sure.
So do you think they're both representative of this area, or do you think that, I know in some places we see rocks that maybe were not necessarily from this area, maybe were ballast from ships at one point, or were somehow introduced in other ways?
Yeah, I mean, that's a great question. I don't have a great idea of where they could be transported from locally, I guess. Just because I kind of assumed when we kept seeing these rock cobbles that we would kind of approach like a steeper slope, but perhaps that steep slope is just further away than what we've reached. But given the geologic past of this region, I would say this is likely glacial erratic that's just kind of been dropped off, or yeah, kind of what you said, just kind of a random assortment of rocks. But it is really interesting to note their like angularity and how like rounded and weathered a lot of them look.
So would you say this is similar to like a moraine field on land?
Yeah, probably. Yeah, exactly. Yeah, that's a perfect word for it.
So potentially rocks that were carried by glaciers from a different region, and then as the glaciers melted, dropped off these rocks that aren't necessarily part of this landscape, but now have become part of this landscape.
Yeah, definitely. That's what I would say, especially since so much of these submarine canyons, such as Nitinat Canyon, have been formed by those glacial retreats. So yeah, I would say that could be really likely, but also that kind of highlights the importance of taking the rock samples that we do, because we are able to take a closer look and see, okay, could this be true glacial erratic? Is this some kind of different rock that likely was not formed nearby? But I would say, yeah, given the location, that would be a really great assumption.
Okay.
Yeah. And Jenny Waddell, who works for the sanctuaries here, put an interesting note into our side chat here, that near Quinault Canyon, they often find Australian coal from a shipwreck.
Oh my gosh.
From a long ago past shipwreck. And that was a really big challenge for them. They had an expedition hunting meteorites that fell in the sanctuary in 2018, and they found more of this Australian coal instead.
Oh my gosh, that's so random. Yeah, I guess it doesn't take a glacier. It could also just take humans.
I think it's so fascinating that you can just track kind of like the story of a rock in a way.
Mm-hmm.
There's something about that that's really fascinating to me.
Yeah.
You kind of overlook rocks in day-to-day life, especially if, gravel parking lots and things, but each of those rocks have a story, how they ended up where they are, and it's not always kind of like very obvious what that might be. It may not be linear, and it may not make sense. So,
yeah, I
great story,
definitely
Jenny.
agree. Yeah, I definitely agree. That's one of the main reasons why I love geology. You know, there's always, there, like you said, there's always a story with a certain rock and its formation and how it's weathered and how it has kind of evolved throughout time. And another thing I also love about geology is how when we think of geologic time, we're thinking of like millions of years, which is just so like amazing to think about our Earth on such like a major scale like that.
So, Paige, could you maybe give viewers back home, like, we collect these rocks and then they go to the repository at OSU. What happens with rocks? What do geologists do with those rocks? Do you have any ideas?
Yeah, that's a great question. So, well, I'll first probably add that I am a geophysicist, so I don't work with like hand samples very often, but I am familiar with rock samples being in the geology department at Western Washington University, and there's a variety of different things that you can do with rock samples. I would say the first thing is definitely IDing what kind of rock they are, and usually that's you know, it's I don't want to say a hard process, but, you know, rock environments are so diverse that often a rock sample could kind of be a culmination like of a bunch of different factors, and so you really need to look at the mineral assemblages through a microscope, and also kind of looking at the different like patterns that you see on the surface that could be indicative of its formation when it like was a not even a hard rock yet, when it was just kind of like a
mud.
Yeah, it could be mud, or it could be like a plutonic body, just something like plastic that had to eventually form. And so, yeah, oftentimes when we see a geologic sample like that, that's kind of our first mission, understanding what kind of rock it is and what it tells us about its environment around it. You can also look at patterns of rock too, not just like one rock sample in particular, but like a kind of sequence of rocks. There's in marine geologic environments, there's ophiolite sequences that kind of are indicative of the oceanic lithosphere, which is kind of a fancy way of saying the upper mantle and oceanic crust that make up the oceanic lithosphere. And there's a very particular sequence of rocks that you see in the ophiolite sequence that are indicative of this. And the reason it's really cool is because in places like offshore Washington, there are like some regions where the oceanic crust has been like completely uplifted, and those different layers are completely like vertical almost, and you can like track them kind of along the coast. So it's pretty cool. Yeah, I love all the different sort of things you can just get from one little rock.
That's awesome. Thank you.
Yeah.
Yeah, it kind of looks like it.
Something floating, yeah.
It's something.
A ghost.
It's a thing. It is kind of interesting how we were in that really kind of rubble field, and now it looks like it's. I'm not seeing any of those big rocks anymore. It's a lot more like sandy, if that would be how you would characterize it.
Yeah, I would say it's a little bit more silty, a little bit more fine-grained. But yeah, definitely those boulders are gone, and even though we haven't had the chance to look at like sheer rock faces that are exciting because they host a large biological diversity, it is kind of nice to get a sense of the landscape, and in that way we're kind of like traversing up a slope and then descending back down into like a little valley and then back up, and this is just a nice way of kind of checking our bathymetry and getting a sense of what this environment is like. So in that way it is still a great exploration dive.
Kind of felt like we were going up almost a ridge line there for a minute.
Yeah, definitely. Yeah, just hiking through Nitinat Canyon.
Yeah, good eye. This
definitely
is a sponge. It's the most structure around here right now.
Yeah. Seems to be holding on for its life on a rock.
So I wonder, Ashley, if you could maybe answer this. So I know, like, if you plant a tree in an area that has a predominant wind, then the roots become stronger for that tree. Can that thought process be extended to sponges or any other corals or anything that would append to the seafloor or attach to the seafloor?
That's a very good question. I'm not sure if it makes them stronger necessarily. Maybe. There are definitely some species that you'll find in heavier current, which suggests like they're better at holding on to areas or better at using the food in the water column that drifts really quickly by them. Mm-hmm. You will see like some sponges and some corals will be almost, they don't have as hard of a structure as a tree does with its bark necessarily, but you will see sometimes that they have almost like a bent over posture sometimes. I think that might be just an artifact of like the currents currently going by and bending them though. But yeah, that's a really good question. I don't know if they make harder holdfasts. But yeah, and this sponge looks like one of the ones we collected yesterday, so it does look like a type of glass sponge. And it does have a crab inside of it. Mm-hmm. It also has a hole down on its
oh yeah, it looks
inside
like
of it.
Punctured kind of.
Yeah,
2 holes.
Yeah. So interesting. Might have been a good meal for somebody. Yeah, I agree, Jenny. Not doing so hot.
Yeah.
Sponges are only supposed to have the one large hole that which is where they have water come in, and then they have many small holes that we can't see right now.
Yeah.
They aren't meant to have those other holes on the side.
Do you know what that like mucus kind of near that?
Yeah, it kind of looks
is?
like a salp, which is normally a water column organism. So probably again, this crabs tend to be kind of they'll eat whatever they find. They're generalists a lot of times. And so my guess is that this was drifting through the water column, and you can see it's got a lot of particulate on it. So it probably was already sinking down, and this crab found it and is now possibly going to make it a meal, or it's just stuck to its claw. I can't tell if it's actually going to eat it.
True. Yeah, and Kelly was asking, and I was wondering the same thing. Could it have been damaged by the crab?
Yeah, I mean,
I don't know.
It looks like it, maybe. That kind of looks like that hole might have been there for a little bit, just based on how much sedimentation there is around it, but I don't know for sure.
Yeah. Yeah.
Yeah, it's really interesting. And in a lot of these ecosystems, we aren't down here enough. We don't have enough eyes on the scene to really get a really thorough grasp about all of the interactions between all these animals. So that's why it's really exciting when we're able to see predation events happen on our video feeds live, because it gives us a lot more information. Otherwise, it's kind of, we don't know what was necessarily eating this sponge. Sometimes we know what these animals are eating, but a lot of times we don't know about a lot of their behaviors. That's pretty cool. Is that is there a little pink coral colony
on the
in
right?
the background in the right? I think we just did a shift change, so I might have.
Oh, yeah.
Actually, so I have a question about the sponge. Since this one does have some damage and some holes where there shouldn't be holes, do they regenerate? Or once it's damaged, is the sponge kind of just damaged from that point? Does anybody know?
That's a great question. Wonder if anyone in the science chat knows.
So I don't think they necessarily regenerate. They will a lot of times reproduce. Here, give me 1 second. Let me see. Pilots, is there? Oh, okay, never mind. Sorry, I was just trying to make sure we didn't need to zoom on that coral. Back to the sponge though. Sorry, for viewers at home, we are in a control room that has many moving parts going on, so if we get distracted sometimes, that's why. But yes, sponges oftentimes will reproduce asexually. So if a piece gets broken off, then sometimes they'll just grow out of that. So they do asexual budding. I think there's a SpongeBob episode that talks about that. So, highly scientific, very educational. And Let's see. And yeah, and I think we'll see if anybody else from the science community on our science chat has any better answers. We do have some much more knowledgeable sponge biologists that have been tuning in. So this is perfect. Thank you, Abby. So Abigail Powell's on, and she says that studies on shallow demisponges show that they have an amazing ability to regenerate, and they regenerate faster than they grow.
That's
interesting.
That's cool.
Interesting. So, Abigail, do you think that this sponge has the potential of closing those holes?
So this
if
is
given the right nutrients and everything that it
yeah,
needs?
and that's also interesting. So Abby said that the studies are on demisponges, which
this one's a glass sponge. So
yeah, so that's
huh.
also interesting. I wonder if glass sponges also have that capacity, or if they don't.
Fascinating.
And then I think if you're trying to move, Alexa said we've already seen this same coral, so if you guys need to get out of this area, then you don't have to zoom on it. Yeah, thank you. Thank you.
Oh, that's interesting. Abby was saying that some of those coral studies that we mentioned were looking at barrel sponges that were severed by cables in the Caribbean.
Interesting.
Interesting. Cool, that sounds good. Oh, interesting. Looks like we got some holes in the sediment that were kind of eroded away.
Looks like a crinoid.
Oh, yeah.
Center, that orange yellow.
Of course, I was just focusing on the rocks. I
No,
thought that's why we
okay,
stopped.
You're the geologist.
It is interesting to see that structure, though, especially when then we can see like some of those darker rocks as well that are poking through.
Oh, that crinoid is so pretty.
Oh, this is really pretty, yeah.
Yeah, and again, really indicative of how strong this current is. You can just see it whipping around.
Yeah.
Very cool. Thank you for that zoom. Yeah, and there are some, like, here's a good example of those two different colors of geology right here.
Yeah, those two different rocks. That dark one looks pretty cemented, while that lighter one looks kind of unconsolidated.
Is that another, would you think, unconsolidated mudstone? Not this
I
one,
don't
but
know.
the other.
Yeah, this one definitely is a little bit mysterious. But yeah, the other one's definitely unconsolidated kind of mud.
That doesn't look like the colorations from weathering either, though.
No,
this
yeah.
one's a curious one.
Yeah, this could be more erratic. That's just from a different spot. But can't be quite sure until we take it back.
Do you think that one would be worth sampling?
No, I think we already got one glacial erratic. I would love to try to find a rock that's more cemented into the landscape, but I don't know.
You mean to say you didn't want one of those large boulders?
Yeah, just kind of pick one up. It's easy.
I know that the arms on D2 are very strong, but I don't know that they're that strong.
Yeah.
Also be. Is that a sponge on that rock down there?
It looks like a coral, actually.
Oh, yeah.
It is kind of flowing.
Yeah, once again, these creatures just find any sort of rock to latch onto.
What is that, a black coral? Kind of looks like what we collected yesterday.
It looks like Jenny's saying it's a peristenalia.
I think that was, yeah, this is some sort of black coral.
So, Chrysopathes?
Yeah, so Alexis is standing behind us confirming that possibly a Chrysopathes. We collected one of those yesterday. Yep, so it's type of black coral. We haven't seen a ton of corals on this dive, so it's kind of cool when we do see them pop up. Thanks for that, Zoom.
Woohoo!
Yeah, that Chrysopathes black coral yesterday was quite mucusy, so fun to sample.
Yeah, it's my favorite thing when we've gotten those bio samples back on deck, and I've always wondered, what these things feel like, and so it's been really fun to, with my gloved hand, to pick it up into the bio box.
And that is something we do use that as a description a lot, so I do encourage scientific poking of specimens to tell texture.
Sweet.
I think I've been surprised at how different the textures of all the sponges we've been collecting has been as
Yeah.
well, because I've always just assumed when snorkeling and diving I don't touch the corals or the sponges, so this has been kind of one of my limited chances to do that, and they're not all squishy.
Right, yeah, how did we describe that
One was
yellow
crunchy.
sponge yesterday? Yeah.
I can't remember
Crunchy.
which one that was. I think that was the demo sponge that was crunchy.
No, demo sponges are not usually as crunchy. I think both of our glass sponges tend to have a much more crunchy or fragile structure a lot of times. So there was one that the Faria sponges, I think, kind of are like a potato chip, but softer a little bit. And then we had another glass sponge, I think, is the one that it is crunchy.
Yeah. Some of the other glass sponges were almost like fiberglass, woven fiberglass fabric.
Nice sea star in the sediment. Oh, hello.
Looks like another one of those eel pouts that we've been seeing a lot, hanging out in the water column and zipping around. There he goes again. So this is a nice anemone.
I think we've been seeing these kind of scattered around during our dives. Again, super strong current.
Yeah.
Just blowing them.
Really blowing it around. You can even see how strong the currents are. You can see the ripples in the sand.
Oh, that's
silt.
pretty.
Yeah. Which also shows that there's not a whole lot, or the currents are whipping around so quickly that if there was any bioturbation, so biological organisms leaving tracks, it's getting erased right away.
They just get to zoom.
Yeah. Pretty interesting. There is one, it looks like one of those umbellula sea pens on the bottom.
Oh, yeah.
Bottom left. So you can see how it's one of umbellula, those sea pens, kind of on middle left right now. They're one of those few coral that seem to be able to anchor in soft sediment, and they don't seem to mind strong currents as much as a lot of these other little corals do. But yeah, not a whole lot else here. Got one sea cucumber, got a few fish.
Yeah, pretty peaceful sort of slope here.
Yeah.
The currents even seem a little bit less than earlier.
Mm-hmm. Oh, you can see the tube that it's coming out of. That's cool.
Yeah. That's a great shot. Yeah, you can see. And sea pens are interesting. You can kind of see that that white line down the center of it. I don't know what to call it. the body of it, I guess, and it is a harder substance than the rest of the polyp and the rest of the body. Cool. Awesome.
So can they fully retract inside that tube?
I'm not sure. I don't know that they can.
Oh, this is cool. Chris Moss saying that he didn't know the umbellua had a tube.
Yeah, I didn't. I've never seen that either, really. Which is kind of interesting. Thanks, guys. That was a great Zoom.
And for those of you who have been kind of following us, we started at the base of Nitinat Canyon at about 990 meters of depth, and right now we are at about 784 meters depth, and we traversed kind of over this steep slope, and then descended kind of what seemed to be back into another little valley region. And right now the ROV team is just working to try to find another place within their Blueview sonar system where they can see substantial relief. So right now we're just kind of exploring.
Yeah, a couple more of those sea pens right here too, which is interesting. It seems like now that there's not as much of a slope, they're maybe a little bit easier to anchor.
Yeah. It's cool to see the connection between the, I guess, distribution of the geology, like hard versus soft sediment mostly, and how that kind of creates habitat for different types of species. It seems like those sea pens in particular, we see them a lot in the soft sediment because they're able to anchor.
So we did have some scientists in the chat. Steve Oskovich was online and said that the umbellulas that we're seeing, so these sea pens, are not known to have any tubes. So it's interesting that that one looked like it was growing in an area that had a tube. So possibly an optical illusion.
Yeah, let's do it.
Yeah, if possible, that'd be great.
Could have it burrowed in a empty shell, like a worm casing?
Maybe.
Interesting.
Yeah, and this is more. Yeah, it could just be the way that I mean, I wonder if the current is just
the way it's
pushing up
settling
against the base enough, and then maybe the current pushed back a little bit. It was able to move positions, and it just looked like there was a tube around it.
Yeah,
Cool.
maybe so.
Yeah, interesting.
Hmm.
Yeah, this base is much more much more what we expect for Umbellula. Yeah.
Every time I read that word, I see umbrella. And it takes everything in my power just not to say umbrella.
It's more fun than umbrella.
It is more.
Umbellula.
Umbellula.
It's a good one. This is a great shot. You can see the polyps for and I think, I'm not positive, I think those white dots might be parts of gonads. I'm sure that, Steve, maybe you could correct me. We've got a hagfish in the background again.
Oh, neat.
Thanks for that zoom, guys. Yeah, this looks like a different type of sea pen. So we've seen a couple of these as well throughout this dive.
Oh, yeah. I remember seeing these at the start of the dive and kind of throughout.
Mm-hmm.
It almost looks kind of broken at the top.
And Steve is saying this looks like, just a minute, I have to wrap my head around this and not sound terrible. Balticina? Balticina is what we're going with. Hopefully that's right. Formerly known as, oh no, this one's worse, Halopteris?
It's not hard. It's not easy being a scientist.
Here all week to amuse you with, here all day to amuse you all with us stumbling over scientific names.
Oh, another soul.
Yeah, and so now we're in a shallower depth range. This might be a different type of flounder. Again, flounders and soles are very closely related. I'd have to brush up on my IDs. There are a couple of different species known in this area and at these depth ranges, though.
Which, for the folks who are listening, we're at 780 meters in depth.
Super cool.
Yeah, so when we were in deeper waters, we were seeing some deep sea soles earlier today and yesterday. And this is, I believe, a different species, but I'm not positive on the ID right now. I'd have to go dig up some keys.
Beautiful pattern.
Yeah, the flounders have some really gorgeous coloration, and then a lot of people know of them as one of the fishes that is able to camouflage into its background. So the modeling pattern definitely would help break up its silhouette a little bit against the sand here. And this is a good shot of both of those eyes on one side of its head.
Huge.
Yeah, and so that the other side of this fish will be white. There's no pigment on the other side of them, or if it is, it's not the same coloration on both sides. And then no eyes. So flounders and soles are very interesting fishes that start their life off much as we think normal fish would. They swim in the water column like normal fish do, and then they transition. Eye on each side, yep. Swim in usually at shallower depths, and then as they age, they'll start to settle out and become closer associated with the bottom. And one of those eyes will migrate from one side of the head over the top of the head onto the other side, and the fish will start to gradually kind of stop from swimming upright and will kind of lay down onto that side that no longer has an eye, and then once they're adults, they look like what we saw. So they're a very weird life history.
That is very weird.
A whole new definition for settling down as you age.
Pilot, this is Watch Lead. Could we fire Niskins sometime soon off bottom? We could go ahead now. But it would be great if you could come off bottom a little and try to dust off some of the sediment.
And for viewers at home, we've been taking water samples every dive this expedition, and those water samples get used for eDNA, so that's environmental DNA. So we filter the water, and we're looking for really floating bits of DNA from the water column. So that can tell us what sort of organisms are living in the environment, and gives us a little bit of a different way to see without necessarily having to have full eyes like camera on the scene or taking full collections. So that's a pretty cool new methodology. We've been testing out some different methodologies than what we typically use with Okeanos Explorer. So Alexis has brought some very cool technology to pump some of these water samples faster.
Sweet.
So for the folks at home just listening, we're coming off the bottom to take some water samples. We're just making sure we don't have too much sediment on those containers so we get a really good water sample. And then once we bring those samples on deck, we'll take those and process them and try to use them to get some eDNA, which is just our way of trying to in situ figure out what species are kind of floating around in the water column. Would you say that's an accurate description, Ashley?
Yes, yep. Yeah, and I like eDNA. I kind of think about it like how we're always shedding pieces of our hair or like flakes of dead skin cells. All of the organisms in their environment are likewise shedding parts of their skin or mucus or whatever as they're living their lives. So getting some of that water and being able to look at the DNA that is suspended within the water can really give us a good idea of all of the different types of organisms that are living here. possibly beyond what we're actually able to see, because we have big lights and we're really noisy. And so some animals don't even come near us. You can kind of see them sometimes dancing around the shadows in the Sirius cam, and we wouldn't ever see them through D2's camera, but we might still pick up some DNA from them in the water samples that we collect. So it's a really interesting way to try and get a good idea of what's in the environment that maybe we weren't able to see even.
A rock!
And then there was a rock, a single rock.
And some interesting holes, too.
Looks like a bunch of worms on it by chance.
Oh, yeah, we've seen these. I forget what those are.
Those are some sort of worm that builds tubes. So they're...
Sorry, I didn't mean to interrupt you. And that sea star on the left is a Hippisteria.
Yeah, so we've seen a couple of those today.
Is it a sponge underneath the rock, kind of on the side?
It might be a tunicate. Scallop.
Yep, scallop.
It looks like one of those scallops we collected yesterday.
Yeah.
Cool.
Have an iridescent color.
Yeah.
That just kind of closed a little bit.
Oh, and those little worms are called supulid worm tubes.
Yeah, and Chris Ma is pointing out so that that Hippisteria that we have to the left of that rock is kind of arched up a little bit, so that's indicative of it feeding on something right now, as opposed to when we see them just kind of sprawled out flat on the sediment.
Interesting.
Yeah.
Is it a burrow in the bottom center right?
Yeah, there's a lot of holes in the sediment, which there's so much living in the sediment in these areas that we can't see worms, amphipods, lots of what we call, like, there will be in fauna, so things living in the sediment, that if we were to take like a sediment core or like a box core of the area, then you could capture an idea of what's there. But yeah. Yeah, sounds good.
Perfect. Woohoo!
Thank you. Oh, yeah, I see a shrimp. Yeah. Some of these shrimps are so brightly colored red that they really stick out, especially when there's nothing else around.
They stick out in the light, but it makes them appear darker in the dark.
Yes.
What a cool magic trick.
Right. And so what Caitlin's referring to is that a lot of the animals that live in these deeper depths will be a dark red color, because red is the first wavelength of light that attenuates in water. So it's no longer visible to most of the other animals that live at these depths. So instead of trying to turn dark black, it's easier to just, or can't say it's necessarily easier, but we will see a lot of animals that turn bright red. It essentially turns them black to all of the other animals living around them that are unable to see red. That's pretty cool.
Oh, sweet shell, yeah. No trail behind it.
It is interesting. Maybe it's just a shell then.
It is kind of buried.
Oh!
It does look like it has been grown
Oh,
over
interesting.
by, that's interesting. By something.
Yeah.
Yeah. Open to
Yeah, possibly a lot of worms.
Yeah.
Maybe. Oh, it is still alive, though. You can see the siphon coming out.
So maybe it's feeding on something?
Yeah,
But
and it's not uncommon to have things kind of grow on shells, but this is interesting to see this much coverage. Yeah.
Are those hydroids on it or worms? What do
It
you guys
doesn't look
think?
like hydroids to me, but
Chris Ma says he
it looks
thinks
more
it might be hydroids.
Oh, okay. Maybe it is, yeah. Yeah, they aren't really branching, but hydroids also take many forms, so. And then I think whenever is a good time, we can also fire the other Niskins after we get off the bottom a little bit, too.
Ooh, what?
Oh, but you sounded like you guys were excited about something, so that sounds more important. That sounds more interesting.
I put sonar up on stream 3. See the line of holes the pilots are aiming at?
Oh, that is interesting. Yeah, it's cool that you can see that in the blue view. So for viewers at home, we have a very nice sonar on D2. It's called a Blue View. And the pilots just saw like some very interesting depressions, it looks like, in the sediment that we might be coming up on soon. So they're heading in that direction to see if we can see anything. Yeah, it is very much like a couple of lines of depressions.
We're on the move.
The depressions look so slight, I don't even know if we'll be able to see them from the camera view.
Yeah, it is
The main
interesting.
camera.
Sometimes we can see more things like in a, depending on how Sirius is oriented, sometimes you can see more from Sirius than D2 depending on how the
Clear the water
features,
column is.
yeah, and like what the features orientations are, things like that.
So I guess this is one of the depressions now that we're seeing. Oh.
Yeah, sounds good.
Thank you.
Sweet. Oh yeah, another shrimp and a sea cuke. Little crab.
So we got a nice little shrimp here.
Oh, pretty color.
Yeah, very interesting coloration.
Yeah, it is an interesting. We usually see these are more brightly colored red, and so this one looks a little bit like a yellowish color, which is interesting.
Yeah.
Kind of looks like it's been highlighted.
It does. This one was important. Yeah, so I'm not sure exactly on an idea. We have been seeing a lot of shrimp in the family Pandalidae. Could be one of those. But I am not a crustacean biologist, so I'm not positive on the ID on that. There was just a little worm, a little feather worm that darted back into the sediment too.
He's scared of the shrimp.
That's what he's scared of.
Not D2.
Not us. Just crawling along in this high current area over here.
Oh, cool. Like 5 centimeters, 6 centimeters.
Yeah.
Yeah.
Oh, and there's another off to the right.
Yeah, same coloration.
Yeah, interesting. Be curious to find out what that is. I'll have to ask some of our scientists ashore that are experts in that group of animals. No, I don't think that they're of interest for collection necessarily, but thank you. I can right now. Niskin bottle? And it looks like we've got a skate coming into view too.
We got a nice skate here.
Yep, so this looks to me like
a male.
Yep, so it's definitely a male. It looks like it's, I would guess, so genus Bathiraja. I think it's Bathiraja trichiura, which is I think it's called deep sea skate is the common name. So we've seen a couple of these guys along this dive. I think they're really, I love the dark coloration on them. They're very pretty, but. I can answer that question. So they.
Oh, do you want to repeat the question?
Yeah. So the question for viewers at home, Roland on video asked the question of what's the difference between a skate and a ray? So they are closely related. They're both, and they're cartilaginous fishes. So they're also closely related to sharks, but they are in different groups. Skates do not have that stingray that stingrays get their name for. So they don't have a venomous barb on their tails. They tend to have these thorns. So all of these thorns are denticles. So we have thorns and denticles. They give all of these cartilaginous fishes kind of a like sandpaper-like consistency.
Oh, really?
Yeah, so the thorns and skates are actually are used to identify the species. So that's really important for trying to determine which species you're looking at. So you can see on the wings, too, how there's like that lighter coloration. Those are a bunch of thorns as well. So that's really important to help identify. A lot of shallow water skates will have thorns all over their body and up towards their eyes. And how big those thorns are, where they're located, those are all used to identify species apart from one another. Stingrays don't really have those, and they really just have that venomous barb coming off their tail. We don't see stingrays at these depths. You'll only ever see skates at these depths. Stingrays tend to be much more shallow, and we also find skates shallow, but they have a much broader depth range, and we'll see skates at these depths, but they also don't extend as deep as a lot of fish species do that are bony fishes. So we were seeing a lot of different fish species when we were really deep on our dives earlier this expedition. We've only been seeing the skates on the last two last two dives where we've been shallower. And again, shallow is relative. We were still around 1,000 meters, so still deep.
Currently at 762 meters.
So this is a great view. So those are spiracles. That's how the skate will pump water over its gills so that it can get the oxygen it needs. And then their mouth and they'll have their mouth on the bottom side of their body, so we can't see it right now, but they glide around much in the same way that stingrays do, where they're finding food in the sediment. Unlike other cartilaginous fishes, they do have an electrosensory system, so they can detect small vibrations that can help them find prey in the sediment as well. This is a great zoom.
Yeah, you can see some of the ridges.
Yeah.
Oh, yeah, those barbs.
The textures on the barbs.
Like scary.
So, and these guys do look like they should be soft sediment, or they should be soft sediment. I'm thinking too much about geology right now. They look like they should be soft to the touch, but like many of the other cartilaginous fishes, all these, you can see the texture now that we zoom in. So they've got these denticles, so they do feel much more like sandpaper along that.
Oh, cool.
It's a great view. Yeah. So, and again, we can tell that this is a male because it has claspers. So those are the two kind of appendages that you see next to its tail. So those are the sexual organs. Males have claspers, females do not. And in a lot of skates, they're very prominent, and you can see them very easily. So that's it's good for fast ID on if you're looking at a male or a female. The females will just have the tail and won't have those two other appendages off to the side.
And since it's related to sharks, sharks will also have those claspers as well.
Yes, yep. Sharks, skates, rays, and chimeras all have clasper sexual organs. Yeah, that was great.
Yeah, that was cool to watch it move.
You. Yeah, thank you.
Oh, Jenny Wandell, who's from Olympic National Marine Sanctuary, has said that they've also seen long-nose skates on previous dives in this area.
Yeah, and in the shallower waters, there are several species along this area. But yeah, depth is a pretty good indicator of some species, especially for skates. Many species don't come this deep.
Sea stars.
They always look so much larger through the camera. I'm very curious to see how large the sea star we collected earlier will actually be in the lab.
Mm-hmm.
It's also interesting because when these animals live down at pressure, sometimes when we bring them back up to no pressure in the wet lab, they are not necessarily the same size that we thought they were down here. It's interesting to see how they don't look the same. Kind of like blobfish does not really look like it's frowning all the time in its natural environment, but when you bring it up to zero pressure, it gets a little bit.
Oh, does it get wrinkly?
It gets wrinkly and it can't support itself over the weight of its body.
Yeah. Could we get a quick zoom on that sea star?
Awesome, thank you. Oh yeah, I didn't even see that. Yeah, there's one of those Umbellula sea pens right at the bottom left of the frame. Possibly. It might be that.
An anemone.
It might be an anemone to the just left of the sea star. So we'll see what.
Oh, cool.
Pretty spiky.
Yeah.
Yeah, really white.
So Chris Ma is in the chat, and he said that it's a Zoroaster Ophiura. Ophiura? Yeah, and there are some, it looks like, polynoids associated with it.
Yeah, it looks
possibly.
Like 2 of them potentially.
Yeah,
one on the forearm and then one
Oh,
on
hello.
the back arm.
Yep,
Hello,
and
friend.
there's a little eel pout.
That's cool.
Interesting.
Yeah, great zoom. Thank you, guys.
Yeah, thank you. Whoop.
Perfect, thank you.
Sweet. I think we still got one more Niskin.
We do.
Is that a rock in the distance, I wonder?
Oh, so that's interesting. Chris Ma was saying that they have, pediciliaria? I'm not going to be able to pronounce that word. I'm sorry, Chris. But they have spines which they use to dig into the sediment and feed. Very cool.
Pedicellariae.
That
sounds right.
Yeah, we'll go with that.
Oh, that's cool, though.
Goodness, Paige, is that a rock?
Ah, that is a rock. Yeah, that is a nice rock. That's cool. I haven't seen a rock in a while, actually.
No.
A lot of sediment here.
Yeah, after that boulder potential marine field, it's just been pretty sandy, silty.
Mm-hmm. Ooh, and a sea star on the side of the rock like we've been seeing.
Mm-hmm.
Don't know why they do that, but it's interesting to see and observe here. Oh, whoa.
Geology and biology biz.
It is really cool, though, how these like animals are able to just find like one of the few surfaces that there are in like this valley.
Yeah, they've got a housing crisis down here too.
Yeah, for sure.
They really need to take up every little piece of real estate they can. Yeah, so what we're seeing is this rock in the center of you is just covered in these worms that build their tubes. And it looks like it might have some other associates with it as well. As we zoom in, I'm sure we'll find more stuff. So there's a little, looks like, oh, can't tell, maybe a benthic jelly even to the bottom.
Yeah, I was wondering what that red jelly was.
Yeah, it looks like there's some jellies. Yeah, so you can see these worms, they have this little feathery appendages. They stick out into the current to catch particulate drifting by so that they can feed. Awesome. We've got some bryozoans, which are the little white patchy areas that kind of look, I don't know, they look like sandpaper to me. I don't know how you would describe that. Those are bryozoans. There's a little, looks like maybe a mysid shrimp up at the top of the rock. And then we've, of course, got hydroids, which grow on a lot of things in the deep. Awesome. Thank you.
Yeah, and so like I mentioned, we've been seeing those sea stars that kind of mount onto the side of these rocks, and the scientists in our science chat have been kind of mulling over what the reason for that could be, because it's a little bit of a mystery, but Chris Ma, who's one of our experts, thinks that it probably has something to do with respiration, trying to be extended into the current.
Interesting.
But yeah, still not sure.
Yeah, that'd be great.
Whoop. We might need a second here in the back.
Okay.
All right, we're good.
Thank you.
The last water sample of EX2301.
Oh, wow.
Isn't that wild?
That is wild. You say it like that. Geez, it sounds so final, because it is.
Oh, there's a hipposteria, and it's eating on something. Can't see what it's eating on, but it's definitely raised in the center.
He is right.
Yes.
Hippastria.
He's getting too good with these.
Yeah, Lars, have you considered going into biology and becoming an echinologist? Oh.
No.
He made his exit.
He just came in to just, yeah, exactly. He's like, yeah, I know what this is. You guys don't know? Bye.
Yeah, some mucus.
This is great. Interesting. It's interesting seeing where the mucus is starting to flake away off of that arm, and then there's still mucus covering the rest. You can kind of see what it looks like. So Chris is saying that all the fuzz is the papillae, i.e. the gills. Yeah, that'd be great if you can.
Nice.
Awesome, thank you. Thank you.
Yeah, and so if you've been following us through Nitinat Canyon, we've been keeping an eye on the dissolved oxygen levels. When we started this dive at 990 meters, the dissolved oxygen, or the amount of oxygen available in the water column, was really, really low, and we were kind of curious as to how the dissolved oxygen levels would change as we got to more shallower regions, and kind of looking at the dissolved oxygen readings that are coming off of D2, it looks like it's still continued to be low. Right now we're at about 755 meters of depth, and the dissolved oxygen is about where it was at the beginning, but it's at 0.38 milligrams per liter. Yeah, so again, it seems like we haven't really been able to get out of that oxygen minimum zone. Which could also be a reason for the low sort of a species diversity. That, I think, and combined with kind of the lack of hard substrate. And as we kind of near the end of our final dive today of our expedition, just wanted to remind everybody that this is EX2301, Shakedown and Express West Coast Exploration with NOAA Ocean Exploration and the Global Foundation for Ocean Exploration. This expedition has been a really amazing time. We kind of started in off of Astoria, Oregon, worked our way kind of down to Northern California, and then looped our way back up, kind of following the margin off coast. And we've been able to see a lot of really great locations. We kind of examined thrust faulting along the Cascadian Deformation Front. We've been able to observe some uplifted regions within the continental shelf and the continental slope, and just recently we've been just kind of exploring two different canyons, Quinault Canyon off of southern Washington and Nitinat Canyon, where we are now, off of northern Washington. And yes, it's been an amazing expedition. We found a lot of species and a lot of different geological formations. We hope that you've enjoyed kind of following along with us. And yeah, we're still not done in Nitinat Canyon, but just thought I'd remind everyone. Oh, and I also wanted to mention that we'll be having a port event this Saturday, which I always forget the day. It's April 29th.
Yep.
Thank you. And we're the ship Okeanos Explorer, the vessel we are currently on, will be at Pier 46, I believe, in Seattle, Washington, and we'll be there on April 29th from about 10 a.m. to 5 p.m., and that'll be an opportunity to tour the ship and talk with some of the explorers and scientists and engineers here on board. You can also learn more about NOAA and see the ROVs that we use to dive to these amazing depths. So we'd love to see you all there.
Pretty cute little hermit crab we've got here.
Yeah.
Just trudging through all this sediment.
Yeah. He's got a lot on his shell.
Yeah. I think we're giving him a little bit of a wash.
Mm-hmm.
That is interesting that now we aren't seeing those jellyfish anymore in Sirius view. I wonder what depth they stopped being kind of prevalent in the water column. It's interesting.
Yeah.
Looks like we've got a snail, and it's got some associates on its shell, so it looks like there's an anemone.
Oh, we saw something like this when we were at the bottom of Nitinat.
Yeah.
That's cool.
Yep. Oh, yeah, right at the front of the shell. Yeah, it does look like maybe... it looks like an amphipod to me. Yeah, so probably just catching a ride.
Nice.
Awesome, thank you. Yeah, I see a shrimp. I see a little crab. No rocks. Oh, rocks! God, we were just zoomed in too far.
Oh, they're there. They're always there. Yeah, I think we're good on rocks. Thanks.
That's quite the change from the beginning of the expedition where you were collect all the rocks mentality.
Yeah, those rocks are really interesting. It would have been cool if they were more consolidated. But yeah, those ones were really the ones that we previously saw were really embedded into the landscape, and it's really nice to be able to just pluck them out from their exact location instead of just kind of picking up one that's tumbled down, because it's harder to target its location. Still neat to see here, though.
Mm-hmm. Yeah, there's a lot going on here. And so as we're looking at this, looks like there are hydroids just covering every part of this rock.
Mm-hmm. Yeah, making it look fuzzy.
Yeah, so all of these branching kind of organisms that are coming off, they're hydroids, so they're kind of related to other cnidarians, so other jellies and corals that we see, but they're a little bit more simplistic. We've got a bunch of... So some shrimps. We've got a little squat lobster.
Oh, the squat lobsters are cute.
So cute.
Is it?
It looks like it might be missing an arm. Yep.
Ouch.
Yeah. It'll grow back, and it's next molt. Don't worry. We've got some little, looks like some amphipods. Yeah, so these look like some of those pandalid shrimps. Mm-hmm. Looks like maybe some eggs up there in the top right corner.
Oh, cool.
I don't know what they're from, but that's what it looks like to me.
That white-looking stuff?
Yeah.
Huh.
Stuck on that hydroid.
Interesting. We saw some... Were you there when we saw some brooding
I was downstairs.
Shrimp
I
here?
saw, yeah, it looked pretty cool, those brooding shrimps.
Mm-hmm.
Oh, we've got some little capryllid amphipods on the right. They're the little stick-like amphipods that we've seen commonly associated with corals, but they're all over the place. Yeah, this is always fascinating when you zoom in. Just it always looks barren, and then as soon as you zoom in, there's just life everywhere.
Yeah, I really appreciate these sweet cameras that we have on D2.
Yeah. And it looks like possibly a tunicate right above that other squat lobster. We've got some more of those tube-dwelling worms here, making those nice white lines that we're seeing, those little squiggly lines. It looks like maybe some encrusting sponge that we're seeing now on the left right here.
Yeah, they're hiding so much of the rock I can barely tell what it is.
More hydroids. And a fish.
Thorny head.
Yes. It really is fascinating to see just how much life springs up around these rocks.
Mm-hmm. Yeah, it's cool.
Awesome. Thank you. Great view.
Still super sedimented bottom. Super flat as well.
Okay.
Sounds good.
Awesome. Thank you. 11 minutes of muddy bottom. Okay.
Cool.
Cool. Thank you. I feel like sometimes we get a better view in that blue view sensor than we do with what we're seeing. Oh, what is this? Oh, looks like possibly a little shark up in the water column.
That?
Yeah.
Oh, cool.
Yeah, so it'll be some sort of a cat shark. Yeah.
Oh, cool.
Yeah, a little cat shark would be my guess. Cool. Well, that was fun to see.
Yeah, so I would say it's been really exciting exploring the Cascadia margin. This margin is really, really important for kind of understanding the geohazards that are associated here. So we kind of have an interesting tectonic environment offshore Washington, Oregon, and Northern California. We have the Juan de Fuca plate, which is an oceanic plate, and it's formed relatively near offshore, and so it's a relatively young plate, making it hot and buoyant. And along the coastline, it meets up with the North American plate, creating a convergent margin. And this convergent margin is called a megathrust subduction zone, and so it's capable of hosting really large magnitude earthquakes, and we haven't seen a large earthquake along this margin since about the year 1700, and so since we haven't seen all of those earthquakes since then, since modern technology, it makes it really, really difficult to study the margin and predict geohazards in the future. So it makes this work that we're doing here along the surface that much more important for trying to understand those earthquake and tsunami hazards. And it looks like another rock. Hooray. Looks like some sedimentary rock.
Yeah.
Yeah, so some of this rock that just seemingly seems to come out of nowhere is likely called glacial erratic, which means that when glaciers were in this region about 12,000 years ago during the last glacial maximum, as they swept the landscape, so to speak, they kind of scraped up the rocks that it was traveling over, and it cemented those rocks into its ice sheet. And so as that ice sheet melted, it just kind of dispensed these rocks onto the surface. And so that's likely a lot of these kind of mysterious boulders that we're seeing just kind of randomly out of nowhere.
Interesting.
Yeah. Icebergs? That's a good question, because I think icebergs are primarily just buoyant on water, so I don't think so, but I'm also not sure. Yeah, like off of like a... Yeah, definitely. Yeah, less common. Yeah, different ice.
So Jenna Hill in the chat says that usually icebergs or ice rafted debris is usually smaller grain size unless it's really close to the coastline.
Yeah, sometimes along like the coastline, if you're ever like in the shores in like Washington or whatever, you can see just kind of like random pieces of granite rock that are just distributed, and those are like pretty prominent glacial erratic type rocks that you can see along the coast that have larger grain sizes, more like polka dotty.
That's wild to think about, just glaciers just carrying.
I know.
Like that.
Yeah, well, they were like massive. It's actually like really interesting. I teach a marine geology course, and we get to go out to the coast kind of north of Seattle, and you can even see like striation marks where the glaciers left like these scrapes, I guess, kind of along the rock below it, and just totally flattened out the landscape, which is really, really cool to see. And you can even feel the direction that they go in,
oh my gosh,
which is wild.
Geology's cool.
Geology is cool.
Yeah, rocks
You
rock.
could say it rocks. Yeah, Jenna just wrote, and I completely agree with this comment. It might not seem like it, but glacial environments are some of the highest energy environments in the world. Yeah, the power of water, even in ice form, is spectacular. What fish is that?
I think it's
Small.
a cusk eel, but it also could be. There's one eel pout that looks suspiciously like a cusk eel to me, so it's one of those two.
Super shiny.
I think it's we've been seeing a lot of them, and this is the best zoom we've had on them, which is great, so I can go look at my ID guides afterwards and try and get an identification on it.
Nice.
Yeah. And it decided that I didn't need any more of an ID, so I guess that's it.
Just shy.
Mm-hmm.
You guys have seen a couple of these recently, Paige?
Yeah, we've been seeing them all over the place. We actually did a zoom in on one earlier that seemed to be feeding on something. But yeah, the mucousy membrane. Its gills are really interesting, and the spiky on the outside.
I think this is one of these Hippisteria
Californica.
Yeah, so this is, we're getting close to the end of our dive today, and this is the last dive of our EX2301 expedition. We were hoping for a canyon wall with a little bit more hard substrate, but you know, sometimes when you come down here after mapping the seafloor, we only have really like a 25-meter grid to go off of, so we don't know exactly what we're going to encounter. But that's part of the fun and excitement, if you will, of
the exploration.
Ocean exploration is getting to, you know, get eyes on the seafloor, and sometimes you find what you're expecting, and sometimes it's quite a different habitat. And I think Paige has talked really well about the geology and the seismology of this area as to why we might be seeing this type of environment and not necessarily steep canyon walls. But we've been able to observe a lot of different types of fish and other benthic organisms, so animals living on the seafloor, even if they're not our corals and sponges that we're kind of always on the lookout for. Awesome.
All right,
thanks,
sounds
Nav.
good. Thank you.
Yeah, so that was our navigator saying that we have completed this dive. We are out of time, so we'll be headed back up to the surface. Any final words, Paige?
Wow, it's been an amazing expedition. I've had such a great time exploring the Cascadia margin. You know, I live in Bellingham, Washington, so it's been really, really fun exploring the deep waters here. And thanks for our onshore science team who has helped us narrate along this journey and has provided us with their knowledge base. It's really, really been a great time, so thank you.
Yeah, we've had a really amazing group of onshore scientists that have been engaging with us, both to help identify biological specimens, to talk about the geologic features and the history of the area, as well as kind of help us identify what are specimens of interest to collect, both from a general biological, just generally on a biological side, and maybe what's of interest to nearby sanctuaries like the Olympic Coast Marine Sanctuary.
Yeah, definitely.
So we will probably be on surface pretty quick today because we're only at about 730 meters. So the ROVs will work their way to the surface, and then Paige and I and the rest of our science team, Ashley and Caitlin and Jennifer, will process these samples and get them tucked away so that they can be sent to their various repositories when we get to Seattle.
Yeah, and if you have some time, we'll be having a public event in Seattle on April 29th, this Saturday from 10 a.m. to 5 p.m. At Pier 46. And that'll be an opportunity to see the ship Okeanos Explorer and talk to some of the explorers and scientists, and also learn more about NOAA. And if you want to see these deep sea robots in real life, this would be a great opportunity to do so. So we hope to see you there if you're in the area. And yeah, thanks so much.
Yeah, thanks everybody. Okeanos will be back out exploring after a short port call in Seattle, and they'll be headed up towards Alaska. So they'll have a couple ROV cruises this season off of Alaska, as well as some more mapping cruises to fill in lots of mapping data. So keep an eye out on all the social media as well as the website, and you'll be able to tune in for those as well.
Heck yeah.
Everybody have a good life.
Have a great ascent.